big hello to all my wonderful PU2 aspirants, my dear children. I hope that you are doing wonderful. First things first, I feel so happy to see you all comment down under chemistry, physics and math video that the exams were so good. As a teacher, I am telling you, it feels so nice when your students say like that the exams were good or were easy, it was not very difficult or whatever it is, or basically I've just improved myself. All these statements are really very important to us, and seeing you take your time to put down those comments under our videos even after exams is something so exemplary. I'm so thankful for all of you, even though those were not my videos, my subjects, but still I feel so happy because it's you at the end who is the happiest state, which is why you are commenting down. You get it? So your happiness matters to us, and I think I could also maintain the same thing for biology exam. I could also make you happy, okay? And I just want one thing from you. Just give your 100% effort, my dear children. You might be falling in two categories. One category of children who have already completed the chapters. You just are here to brush up your things, and there is another category of students who might have just completed the important chapters until now. There are a few chapters still left to be covered, okay? And you might be feeling that I don't know how do I cover it within this short span. It's okay. It's okay. All of you who are listening to this particular video, you are going to ace biology exam. That is my guarantee. So we have a plan, and please follow that plan. Please read your NCERT, okay, just once, okay? Any chapter, for example, if you're taking sexual reproduction in flowering plants, take the chapter and then come watch the videos. So every video that I have made until date, made until date, basically I have categorized chapter wise. So you get to know that where do you stand after studying each chapter. Okay, so now in this top 50 plus questions that I'm going to show you over here, I have taken 4 chapters. There will be part 1, part 2 and part 3, my dear children. In part 1 there are 4 chapters, again in part 2 there will be 4 chapters, and in part 3 there will be 5 chapters. I have made an informed decision over here to segregate the chapters. I have taken 2 chapters which would be high weightage, low weightage chapter and a medium weightage chapter, such that you don't feel that if I take all the high weightage chapter together, you might feel the burden of it. I don't want you to go through that as of now. So I hope this video is going to be released on Saturday. Please make sure, if you have started watching this video, go ahead until the end, until I tell you that children, you need a break. So I am studying with you. You can imagine like that, okay? So whenever I go on a break, I'll tell you that I am going on a break. At that point, you can also go take a break, cheer up and come back and let's study. Let's make sure that we are utilizing these 2 days. How many days are left? So Saturday, this is Saturday, then Sunday and then Monday. So Saturday we cannot count fully, so I do not say the number, I won't say the number of days which are left, but whatever days we have, let's utilize it completely. So today's day, Saturday, is for this particular video. Watch it completely, it will be really helpful for you. So I am going to take out my time to explain you questions as well. So if you are someone who are finding difficulty while reading the NCERT regarding the concepts, I would be explaining you the answers very much briefly so that you understand things, okay? So that is my job, and I am sure that I'm gonna do that for you guys, but you have to make sure that you have to give your 100% over here, okay? So please follow me. And one more thing I had to say, yeah, the next video would be put down in the morning, and that is Sunday morning, and one more part 3, as I said, top 50 questions we have divided. So the last one would be given on Sunday night or evening time so that you can study properly. Please remember, I have already made all important MCQs and also 2 marker, 3 marker and 5 marker video. Please, if you have time, go watch the 2 marker, 5 marker part, or else probably we would be covering it here itself. But still, if you want to just brush up, put it into X speed and watch it if you have time. But MCQs, definitely you have to watch that video. Very important. I would be solving MCQs live also on Monday, okay, so that you can ask me doubts. But yes, but if you do not want any disturbance, you can watch that particular video as well. Yeah, let's get started. So that's a promise that we are going to make. Children, we are going to ace biology exam. This is going to be the best exam that we have ever gave for biology. Okay, I'm not taking chemistry or physics because it might kill me. Yes. Chalo, let's get started. Over here, first chapter is sexual reproduction in flowering plants. It carries 11 marks, my dear children, and with the concepts that I'm going to teach, I am sure that you're going to get 11 marks totally. Okay, so mention two differences between microsporogenesis and megasporogenesis. What do you mean by genesis? Genesis means formation. So you're talking about the formation of microspores. Okay, and here you're talking about the formation of megaspores. So how do we explain it? First things first. So microsporogenesis, that means you're talking about the formation of microspores, my dear children. And what are these microspores going to be when they grow up? Okay, they are going to be pollen grains. They are going to be pollen grains. Over here, what about megaspores? The megaspores which is there, it is going to be embryo sac. You know what's common between the embryo sac and pollen grains? Both of them has the gametes. So pollen grain has the male gametes, okay, and the embryo sac has the female gametes. So you're talking about the formation of how do they form and how do they form and how they are exactly different. Okay, so I'll tell you a story. So in the case of the male plant, you know there is anther. There is what? Anther, my dear children. And in the anther, there are a group of cells, okay, which forms a tissue which is known as sporogenous tissue. What? Sporogenous tissue. Now, each cell of the sporogenous tissue basically is going to act like microspore mother cell. Microspore mother cell, as in their children will be what? Known as what? Microspore. So this is microspore mother cell, then obviously the children will be microspores. Okay. So over here, the microspore mother cell which is there, it is having the normal number of chromosomes just like any other plant cell. But it is now going to start undergoing a process which is known as what? Meiosis. So meiosis happens in two stages. The first stage, when it happens, it gives rise to two cells, but there's a tragedy. The number of chromosomes, it becomes half. So now it is half the number of chromosomes. You call this state as diploid state and you call this stage as haploid stage. Okay, now what happens? Now meiosis 2 happens. Okay, I told you it happens in two stages. So meiosis 2 happens. So over here again they are going to divide. And you might think, ma'am, again will it reduce? No, there is no reduction going to happen. The cells will be having NN. So this cell divides into 2, this cell also divides into 2. Now I'm going to tell you these 4 things that you see over here, my dear children, they all are microspores. They all are microspores. And if one microspore basically gives rise to pollen grains, my dear children, how many pollen grains are going to be formed over here? 1, 2, 3, 4. 4 pollen grains are going to be formed over here. So after one round of microsporogenesis, how many pollen grains do you get, or how many microspores do you get? Basically get 4, and 4 of them they transform into pollen grains. Now let's talk about megasporogenesis. So what happens? This is happening inside the anther. Inside the anther you have sporogenous tissue where all this is thing happening. Now if you talk about megasporogenesis, where does it happen? So basically, have you heard that in the case of female plant they have ovary, inside the ovary there is ovule. So I'm telling you, inside the ovule, my dear children, there are certain cells which are known as, which are going to form nucellus, a tissue. A tissue. Nucellus is just a tissue formed of so many cells. Just like how you have sporogenous tissue, just like that you have nucellus over there. So over here all the cells are capable of giving rise to microspores. That means all of them are microspore mother cell. But this here it's kind of different. Only one cell will be there which will be acting as what? MMC. Ma'am, again same. Where, bacha, over here it is not microspore. It is what? Megaspore mother cell. It is megaspore mother cell, again having 2n number of chromosome. It will also undergo meiosis 1, my dear children, give rise to 2 cells. How much? The chromosome will become half, right? Right? Now again they will divide 1, 2, 3, 4. Okay? All of them again haploid, haploid, haploid. This is the results of meiosis 2. Ma'am, at the end you are getting 4 megaspores like this? No. What happens is that even though there are 4 megaspores being formed, 3 of them are going to degenerate. They are going to die. Only one is going to survive. Only one is going to survive, and that is known as the functional megaspore. What do you call it? The functional megaspore, as in only one is going to function over here. Those are the sad thing that is happening, but it happens. And this functional megaspore is going to give rise to what? An embryo sac mentioned over here. Got it? Did you get it? Okay, and in this embryo sac you will be having the female gametes being formed. Over here in the pollen grains you will be having the male gametes being formed. So I hope you understood the difference. This thing, it happens in the anther. This thing, it happens in the ovule. Over here, what is happening? Basically at the end you are getting 4 microspores. That means you are getting 4 pollen grains as well. But over here, 3 of them are going to degenerate. Only one is going to be functional. Two differences. If you want more differences, still you can write over here all the cells of the spirogenous tissue are going to act like microspore mother cell, but over here there is only one cell that acts like what? Megaspore mother cell. Okay, so you are asked about two differences. At least please write these two differences, that is microsporogenesis occurs in the anther leading to the formation of pollen grains, that is male gametophyte. Why do you call it male gametophyte? Because inside this only the male gametes will be formed. Okay, now megasporogenesis occurs in the ovule leading to the formation of embryo sac, that is a female gametophyte. Done, we are done with the point. Now it produces 4 functional microspores from a microspore mother cell. Yes, and what do you call it? When 4 functional, we call it tetrad. Okay, 4 of them, tetrad. Now over here, megasporogenesis produces only 1 functional megaspore as the other 3 degenerates. Works? Did you understand this question? Did you also learn something new over here? If you did, do let me know. Okay, if you understood this question, then put a star mark. Let's go ahead. Going ahead with the next question. Complete the tabular column given below with respect to the male gametophyte of angiosperm. This is also a model question paper question. So over here, my dear children, you're asked about the male gametophyte. What is a male gametophyte? I just told you right now. It is what? The pollen grains. It is what? The pollen grains, my dear children. Right? And you are asked about the cells which are there in the male gametophyte. So I'll tell you the structure of the male gametophyte. Listen to me carefully. So there is one round over here. Okay, and in this round there are 2 cells. One cell which is known as vegetative cell, and in this big vegetative cell cytoplasm someone is laying around. Who is that? That is the generative cell. That is the generative cell. Okay, now over here you are asked about the names of the cells. So one is vegetative, other one is generative. Apart from that, you're spoken about the shape of nucleus of the cells. So what is the shape of the nucleus of the cells? Over here, please remember, in vegetative cell, because of this guy coming in, okay, the nucleus which was like round and everything, it has now lost its shape and it has become what? Irregularly shaped. Whereas over here, since it has to, come in between a big cell, the shape of the nucleus is what? Spindle shaped. It is what? Spindle shaped. So over here, what do you write? You write spindle shaped. Over here, what do you write? You write irregularly shaped nucleus. What are the cells? Vegetative cells and the generative cell. Now let me tell you one more information about this, okay, which could give you, whenever I explain you something, please don't think that I am over explaining stuff. I am explaining because I want you to solve the MCQs that would be coming around it. You get it? So I'm trying to explain the concepts behind it so that you get any MCQ which is there, so do not lose even a bit of information that I am telling you. Okay, so over here, the pollen grains has its own, it's a cell, so obviously it will be having its own plasma membrane, agreed. Now outside the plasma membrane there is a layer which is known as intine. It is known as what? Intine. It is made up of cellulose and pectin, okay. It is made up of cellulose and pectin. Now outside this layer there is a very important layer, okay, which is not continuous. Not continuous as in it has small pores. So what is this layer known as? It is known as exine, the outer layer, exit, okay, the outer layer. Exine is made up of one very important thing which is known as sporopollenin. Sporopollenin. You know what? Sporopollenin is a highly resistant material. It can resist alkali, it can resist acids, high temperature, low temperature, everything. Which is why the pollen grains can survive, which is why the pollen grains act like fossils as well. Fossils as in something that does not get degenerated, it stays like that, having the intact shape. So exine is made up of sporopollenin, which is a highly resistant material. All the important MCQs comes from this. Okay, now there are pores. I told you. What are these pores? These pores are known as germ pores. These pores are known as germ pores. Now children, listen to me carefully. Okay, over here this is a pollen grain, right? This is the pollen grain. So now the pollen grain will be having the generative cell and the vegetative cell. This generative cell is the one that will be giving rise to the male gametes. Until, ma'am, how much time have you said that it's going to form male gametes? Male gametes, where is the male gamete? So now also the male gamete is not formed. I understand your frustration. So generative cell has to divide into 2. That's how you get 2 male gametes. Okay, that's how you get 2 male gametes. Now this male gametes have to basically go and fuse with the female gametes. So over here there has to be a pore, a tube that has to be made, that is known as pollen tube, my dear children, from any direction. Now this pollen tube which is formed, it occurs through what? The germ pores. It is formed through the germ pores. And who is going to help? Intine is going to help, plasma membrane is going to help to form the pollen tube. Now what is important is that in certain cells, okay, in certain cells, basically, certain plants, 60% of the plants, okay, in 60% of the plants, It is, okay, the pollen grains are being shed off in this particular stage only. After reaching there on the female stigma, that's when it will divide into two. In 60%. Rest 40% which is still left, in them it have already divided and then it goes and lands over there. So in 60% they land and then divide. In 40% what happens basically is that they already divide and then go. Okay, please remember this 60-40 thing. So 60% of the people are the people who basically go settle down and do their things later on. But there will be 40% people who will like, I have to rush things, I have to rush things up. So basically that is a 40%. You know what? I come under the 40%. You tell me about you. So this is the whole thing that any questions coming off from the pollen grains, I'm pretty sure you're gonna answer all of it now. Now next thing, what is filiform apparatus? What is its significance? So my dear children, the embryo sac which is there, okay, the embryo sac which is there, it will be having certain cells, 3 of them antipodals, okay. 3 cells which are there, they are known as antipodals, and over here you have 2 cells, that is synergids, and 1 cute little cell over here, that is the egg cell, that is a female gamete. So 2 synergids are guiding the female gametes, that is over here, that is the egg cell, and then there are 2 polar nuclei. There are nuclei which is there, okay, 2 polar nuclei is there. This polar nuclei is a part of a very big cell that is known as central cell. That is known as central cell. So how many cells are there in the embryo sac, ma'am? So 1, 2, 3 antipodals, 1, 2 synergids, and 1 egg cell. So 3 plus 3, 6. And then this is 1 big cell, that is 7th cell, that is central cell, my dear children. But how many nucleus are there, ma'am? So 1, 2, 3 nucleus. Each cell will be having its nucleus, right? So 1, 2, 3 nucleus, 1, 2, 3 nucleus. Okay. Now these 2 nucleuses are extra, they are just there. So the central cell, instead of having 1 nucleus, it has 2 nucleus. So I'll say in total there are 8 nucleus, but how many cells? 7 cells only in the case of embryo sac. Now what is important is you're asked about filiform apparatus. So over here there is synergids. Yeah, there is synergids, my dear children. Now synergids have a finger-like projection over it. Okay, what does this finger-like projection do? So basically, suppose the male gamete, the pollen grains have landed and it is forming the pollen tube. It is forming the pollen tube. Now, who is the one that will be guiding the pollen tube and say that, hey, come over here. The male gametes that you're going to carry, please fuse it with our friend, that is what? The egg cell. So, who is there for giving direction and everything? It is the filiform apparatus, which is a part of the synergids, my dear children. So, it is very, very, very important for directing the pollen grains. So, pollen tube. So, filiform apparatus definition is what? It is a finger-like projection present at the micropylar end of the synergid cell. So, in embryo sac, you have 2 ends. Where the egg cell is there, you will call this end as micropylar end. Micropylar end. Always remember, wherever there is egg cell, that is micropylar end, and the other end is known as chalazal end. Okay, so it is a finger-like projection present at the micropylar end of the synergid cell in the embryo sac of the flowering plants, my dear children. And significance is that it guides the pollen tube, helps in directing the pollen tube towards the egg cell for fertilization. This is the significance that it has. Please remember, write the answer like this. So in my 2-marker video, I have not included these questions because these are the questions from the question bank, certain extra questions. But I don't want you to miss out any information, which is why I have included, and I am telling you over here. So please make sure that you are giving your 100%, just like how I am giving. It's not my exam, it's yours, but it feels like mine because you are my children. Okay, so yes. So the question, again, a question can become in MCQ asking what is the direction of the what is the order of cells arrangement in the embryo sac from the micropylar end. So from the micropylar end, if you see it's synergids first, then the egg cell, then the central cell, and then the antipodals. Question can be reframed. Tell in the order from the chalazal end. So it is antipodal cell, then the central cell, then the egg cell, and then last the synergids. Make sure you know about that. Yeah, works? Chalo. Going ahead. Differentiate between chasmogamous and clistogamous flowers. Again, a very important question. So what do you mean by chasmogamous flowers and clistogamous? Clisto means closed. Remember, Cl for closed as well. So these flowers are closed, my dear children. These flowers are closed. Okay, suppose this is a plant and I am saying this plant has both chasmogamous and clistogamous flowers. Ma'am, what? So basically, clistogamous flowers are the ones which are towards downwards. Okay, and they are kind of closed. They are closed, and they have the female reproductive part and also the male reproductive part. Now you tell it's a closed environment, okay, no other pollen grains can come over here and do the fertilization. So what will be happening? Basically the pollen grains from here are going to land on the same stigma, okay, land on the same stigma and do the fertilization or pollination to happen, let the pollination or fertilization to happen. So over here, my dear children, this is obviously always going to do something that is known as autogamy. This is autogamy, where in the same flower, from the same flower, the pollen grains of the same flower is going to land on the stigma of the same flower. This is known as autogamy. This is a mode of pollination. Now over here, since the both are from the same parent, will there be any genetic variation happening? Not much. Okay, not at all much. Is this a preferred method? No. Okay, but no. But what is the good thing about it? What is the good thing about it? Anytime there will be a assured seed set. Every time, okay, what is it? What is seed? Basically a result of fertilization, right? So every time fertilization would be happening and there will be a assured seed set over here. You do not, second thing, you do not have to wait for any pollinators. We don't need bees, we don't need anything, wind, water, nothing. Anyway, pollination is going to happen over here in the case of what? Clistogamous flowers. Now let's talk about chasmogamous flowers. What is the difference between that? So chasmogamous flowers are open flowers, my dear children. So they are beautiful petals being opened up, okay? Now over here, suppose the female and the male part is here. Now from the male part it can land on the female part, but also from the other flowers which are there on other plants. Still, suppose there is another plant over here. From here also, the pollen grains can come and land on the stigma over here. So I'll say autogamy is happening for sure. Autogamy is happening because it's in the same flower, okay? And then xenogamy is also happening. If xenogamy means if this is a plant, this is another plant, between two flowers of two different plants, if pollination is happening, you say xenogamy is happening. You say xenogamy is happening. This is in true sense cross pollination. Now there's another scenario where in this plant only there is another branch, and from here, okay, oh, why is not happening? Okay, from here the pollen grains are landing on the stigma. So it's between the different flowers of the same plant. Again, genetically they will be same. Genetically they will be same. This is known as geitonogamy. This is known as geitonogamy. So in the case of chasmogamous flowers, xenogamy, autogamy, geitonogamy, everything is possible, but over here only autogamy is possible. Works. So chasmogamous flowers are open at maturity, exposing reproductive organs. That is a definition. So always whenever you are making sure you're writing difference between question, first thing, please draw a line. Please make two separate columns for writing your answers in the difference between question. Do not please write like this. Okay, just for spacing things I wrote like this, but you make table and then explain. Okay, so flowers are open at maturity, exposing what? The reproductive organs which are there. It allows cross pollination. That is, okay, you don't need to write allogamy and all, but can also undergo self pollination. That means you can write it allows cross pollination, or it but can also undergo self pollination. In bracket you can write basically geitonogamy and xenogamy is allowed, but autogamy is also allowed over there. All the three type of pollination. Now over here, flowers remains closed even at maturity, preventing what? Cross pollination. Undergoes obligate, that means sure, okay, self pollination, that is autogamy, ensuring seed production even in the absence of pollinators. Example is viola, that is viola and oxalis, and also commelina. Man, is viola, oxalis, commelina example of clistogamous flowers? No, Baba, for both. So you write both. Okay, not as a difference between, you can just write, after you write all the differences, you can put a star mark and write, it can be seen in the flowers of viola, oxalis and commelina. So they have two sets of flowers, one at the top that is chasmogamous, one at the bottom that is clistogamous. Works. And whenever you're writing this, as I said, when you're writing autogamy, xenogamy and geitonogamy, make sure you're writing it properly. If you're not sure about it, do not write it. So allow cross pollination, that is also work, but that also can undergo self pollination. That statement is also enough. Okay, don't have to go ahead with this if it confuses you. Do not confuse the examiner who is going to check your paper. Now list the characteristic feature of wind pollinated flowers. Very important question. So wind pollinated flowers, my dear children, the pollen grains, what do you think about the pollen grains? So they have to move. The pollen grains, it is going to be moving around with the wind, right? So if it is very big, can it move around? No. It has to be light, and obviously it has to be non-sticky. Please use a common sense over here. Okay, non-sticky as in suppose this is a pollen grain and it will sticky. In the wind it will go stick somewhere. No, it will not move where it has to. So it has to be non-sticky, it has to be light, it has to be winged as in there will be certain hairs and things like that which will allow it to move. Okay, now talking about the anther which is there. The anther which is going to release this pollen grains, my dear children, what do you think? It should be down in the flower or should be exposed? It should be exposed, my dear children, so that when it comes it can take the pollen grains also along with it. So I'll say it is well exposed. Talking about the stigma where the pollen grains have to come and land. What about it? It should be what? Large and feathery. It should be large and feathery. Feathery because it has to trap. It has to trap all the pollen grains which are going to come over there. So it is large and feathery. Talking about the flower. Flower, there will be only one ovule in each flower. Why? There are cases where there are multiple ovules in the ovary, my dear children. Do we need that over here? No. Because wind pollination, we cannot assure that it will definitely happen. So why to waste the ovules, extra ovules, you get it? So there will be only one ovule, but the flowers which are there, they are arranged in a manner of inflorescence. There will be small flowers which will be arranged in the manner of inflorescence such that pollination can happen at least anywhere among them. So it is arranged as inflorescence. Now example, corn cob, cotton and the date palm. Corn cob, cotton and the date palm. In the corn cob there is a large feathery things, the picture that is given, the tassels of the corn cob, it is nothing but the stigma style and the things like that. Okay, please remember corn cob, the feathery things that you see, the tassels that you see, it is nothing but the stigma and the style and everything. Okay, it is feathery. To trap what? The pollen grains. I hope this is completely clear to you. And also let me tell you, grasses. In grasses, the main pollination, type of pollination is wind pollination. Please remember, very important. Okay, corn cob, cotton, date palm, grasses, and corn cob, these two are very important examples of this. Works. Chalo, next. The flowering plants have evolved strategies to prevent self-pollination justified by giving 2 reasons. So instead of 2 reasons, I have given you multiple reasons because this question can come in 5 mark also. Okay, so the flowering plants that have evolved strategy to prevent self-pollination, as in they do not want the pollen grains of the same plant to land on the stigma of the same plant. So there has to be some methods. One very important method is dichogamy, my dear children. What do you mean by dichogamy? In some species, the pollen release and stigma receptivity are not synchronized. That means even though there is a flower, and over here there's a female part and there's a male part. Okay, this guy says that yes, I want to release my pollen grains, I'm matured. But the stigma says that it's not possible because I am not receptive yet. So even if the pollen grains lands on the stigma, still pollen germination would not be happening. You get it? So pollen release and stigma receptivity are not synchronized to prevent what? Self-pollination. This is a very important technique. Now protoandry is a sub part of it. Protoandry, that means flower in which anther mature earlier. Okay, protoandry means the androecium. Okay, androecium means the male part. So here the male part is maturing first, but the the female part is not. Protogyny means the flower in which the stigma is maturing earlier rather than the anther. So what is the good thing about it? Other pollen grains can come on land it when they are receptive, because other guys anyway not receptive. You get it? So this is dichogamy, my dear children, very important thing. Second thing is self-sterility or self-incompatibility. Self-sterility or self-incompatibility means incompatible. If I am saying that I am incompatible with you, that means we are not going to match over here. Okay, I am totally incompatible. So the pollen grains do not germinate on the stigma of the same flower due to mutual inhibition. So what has the plant done? It has brought some genetic changes, genetic mechanisms. Okay, this is a genetic mechanism which I am telling you over here. This is a genetic mechanism which is there, my dear children, where basically what happened, due to mutual inhibition, both of them have decided we are mutually inhibited. In this genetic mechanism, it prevents the pollen germination on the stigma. So that is the only example or the only definition of what? Self-sterility or self-incompatibility. You can think it of divorce. It's like divorce, right? Or divorce. Okay. Why divorce? Because it is a mutual inhibition that is being taken over here. It's a totally genetic mechanism. Don't relate with divorce, it's bad. Yeah, let's just study about plants in that sense. Yeah, so this is self-sterility, this is dichogamy. Now there is one more thing, that is formation of unisexual flowers. What do you mean by unisexual flowers? A flower that has only one sex in that is either male or the females. Okay, so in that point obviously you are preventing this. You are preventing this type of self-pollination for sure. You are preventing this for sure. But what about the other flowers? Suppose this flower has a male gamete, the male organs. So this can still land. Still it is self-pollination like thing. Then what will you do? So basically unisexual flowers, if both male and flower are the same plant, just as castor and mace, monoecious, it prevents autogamy but not gytonogamy. So at least something you can prevent, and that is what? Autogamy. In the case of unisexual flowers, now children, listen to me very carefully. Unisexual flowers, okay, that means only in a flower there is only one sex. Now there's another category of a plant which is known as monoecious plant. What do you mean by monoecious? Monoecious plants means they will be having unisexual flowers. Didn't understand? So let me tell you. There is a plant like this. All the flowers of this particular plant, my dear children, they have only one sex, either male or female. So this flower is unisexual, this flower is also unisexual. But if you talk about the plant, don't they have both the sexes? Do they have both the sexes? Yes. So this is an example of monoecious plants. This is an example of what? Monoecious plant. Where you have, in monoecious you have two categories. One is unisexual flowers and the other one is bisexual flowers. Monoecious plant says that in this plant there should be two sexes. Whatever you do, either they can be in separate forms or they can be in together forms. Either they can be unisexual or they can be what? Bisexual. Both of them, they are monoecious only. So you have categorized monoecious plants with unisexual flowers, monoecious plants with bisexual flowers. So in the book, here examples that are given over here. Here, both male and the female flowers are present on the same plant like castor and maize. So castor and maize are like unisexual but monoecious. See, they have both the sexes but on the same plant. My dear children. So obviously over here autogamy is prevented because it is not happening between flowers. But what about geitonogamy? Geitonogamy can still happen. Geitonogamy means between two flowers of the same plant. So this can still happen over here. Okay? I know it's kind of confusing, but please remember this. Please remember, example is castor and maize. In castor and maize, autogamy is prevented, but geitonogamy can still happen. Okay, but geitonogamy can still happen. So this is monoecious plants are not going to work really well. We need to have different type of plants which are now known as what? Dioecious plants. Dioecious plants, because here geitonogamy is still happening. And what happens in dioecious plant is that you have two separate plants, and over here suppose this is the flower, you have the female flowers over here, my dear children, but all the male flowers will be there on another plant. So this is a male plant, this is a female plant. You can tell it male and female plant. These type of plants are known as dioecious plants. What do you call it? Dioecious plants. Okay, they are separate. Now over here you tell me, is autogamy going to happen? No, Baba, pollen grains are not over here in the same flower. Is geitonogamy going to happen? No. In between the different flowers of the same plant also, you just have female plants. Female flowers, what do you expect? Nothing will happen. But what about xenogamy? Yes, xenogamy is going to happen. So the formation of dioecious plant is the best because they assure what? Cross pollination is going to happen for sure. Okay, example, papaya. The example of this is papaya. So if you're asked this question, okay, what is the example of that plant that prevents autogamy and geitonogamy but not xenogamy? It is papaya. Now you are asked the question, what is that plant that prevents autogamy, okay, but not geitonogamy? So it is castor and maize. It is what? Castor and maize, because the flowers are not bisexual, they are unisexual flowers, okay, but it is a monoecious plant. So there will be another sex in another plant, another flower. So obviously from here the pollen grains can come. So geitonogamy can happen, autogamy does not. So you are asked about what is the strategies to prevent self-pollination. So formation of dioecious plant is a strategy, right? It is a strategy for prevent self-pollination, and also unisexual flowers in monoecious plant, that is also a strategy, but not very helpful, which is why I explained it later. So first point goes this, second point goes this, third point you talk about dioecious plants, and then you say unisexual flowers can also be produced, but this prevents what? Autogamy but not geitonogamy. Last point. Okay, if it comes in 5 marker. Now you will be able to answer lots of MCQs with the help of this explanation I am telling you. Okay, chalo. Okay, name the 4 stages in the dicotyledonous embryo development. Dicotyledonous as in, what do you mean by dicots? Dicots has a one that has 2 cotyledons. Has what? 2 cotyledons. So you know what? Male and the female gamete diffuse. Okay, male and the female gamete diffuse. What do they form? They form the zygote. They form the zygote. Now zygote is a single cell, my dear children. Zygote is a single cell. This zygote will again divide a lot to form something that is known as what? Embryo. Embryo. Now there are different stages of embryo. In embryo also there are different stages, after which this particular embryo is going to give rise to the shoot and also the root at the end. So you are asked about the different stages of the embryo, my dear children, and I was talking about dicots. What do you mean by dicots? So I told you the male and the female gamete fuses to form the zygote. But I have already told you that in pollen grains there are two male gametes. One gamete fused with zygote. But what about the other male gamete? Other male gamete, my dear children, it is going to fuse with this polar nuclei that you see over here. Okay, in the embryo sac, one guy fused with the egg cell, synergy helps it. Okay, the other male gamete which is there, it is going to fuse with the polar nuclei. So here, suppose the male gamete was N, haploid number of chromosomes were there, female gamete also N, my dear children. They both fused. What do you got? You got zygote. Tada! N, that means 2N. Diploid number of chromosomes are there. Fine. Now the next male gamete, again it has N. It is going to fuse with two polar nuclei of the central cell. Two polar nuclei of the central cell. Two of them having N each. So N, you get 3N. This is known as triploid. And what is it forming? It is forming something that is known as endosperm. It is forming something that is known as endosperm. Ma'am, what is endosperm? Endosperm is that something that will provide nourishment to this zygote to form what? An embryo. So endosperm forms first and then the zygote will develop. You have to make sure that endosperm is formed first so that it can provide nourishment to the zygote to form the embryo. But now when the embryo is formed, it says that I don't want that caretaker. Okay, I'm the bad person. Just imagine that embryo is a bad person. Okay, embryo says I don't want that caretaker. I will be having my own caretakers. So it says that I don't want the nourishment from this. Instead, I am going to make my own nourishment tissues. And basically this thing the embryo forms is known as cotyledons. All the important things which were there in the endosperm, it is now going to be provided by the cotyledons. So endosperm is totally utilized to form the cotyledons. So embryo axis is there, my dear children. In the embryo, you have embryonal axis which is going to give rise to shoot and the root, and then you have cotyledons which is going to basically provide nourishment to this embryo. Okay. Now, when you have two cotyledons being formed, you call them as dicots. For example, groundnut and everything, chickpea, if you try to break it, you can break it into two. Why? Basically, there are two cotyledons. And in between, if you put them in water, it will slowly grow. Why? Because there is an embryonal axis in the middle, my dear children, that can give rise to root and the shoot, which is there. Now, there are two types of plants. One is dicot. The second one is monocot. What happens in monocot? Here, the embryo is like, okay, let's respect the caretaker. Let the endosperm be there. Let it still provide nourishment. But I want to have my own caretaker as well. So there will be only one cotyledon and there will be little amount of endosperm still persisting over there. That are monocots. We cannot break them down into two. For example, rice and wheat and everything. Okay. Now, so you got an information that zygote and endosperm is being formed over here. Okay. Zygote and endosperm is formed over here. And this endosperm is going to provide nourishment to the zygote to form the embryo. The embryo, when it's in big good stage, it's going to form what? Cotyledons. It can form two cotyledons, then it will be dicot. If it forms one cotyledon, then it is monocot. You are asked about the development of dicots. So where are we? We are here. Okay, development stages of this embryo. Now embryo has different stages, I told you. So first stage is pro-embryo, my dear children, pro-embryo stage. Then there is, so this is the pro-embryo stage which is there. Okay, this first one that you see. So this is a zygote. Okay, now over here the zygote, as you see, it has divided, the cell has divided into two. So this is now an embryo. Zygote is always single celled. If it has divided, it becomes an embryo. So this is pro-embryo. This round thing is globular embryo. Then you have heart-shaped embryo, and then you have what? Mature embryo which is there. So what is the stage? Your pro-embryo, then globular embryo, then heart-shaped embryo, and then you have the mature embryo over there. So these are the four stages of dicotyledons embryo. Even for monocots, you have the same thing. But in monocots, you don't have two cotyledons, you just have one cotyledon, and endosperm is also still persisting. Cool? Works? Okay? Chalo. Now, what is perisperm? Give an example. What is perisperm, my dear children? I told you in this nourishment series, I was telling you all this thing, that basically it's the endosperm that nourishes and things like that. So, listen to me carefully. So, over here, this is the ovary. Inside the ovule, I told you there are cells known as nucellus, okay, which forms a tissue that is known as nucellus. And one cell among them is going to form what? One cell among them is going to form the embryo sac, the megaspore, and then the embryo sac, where the zygote and everything is being formed and everything is happening, okay. Now here the zygote is being formed, endosperm is formed and everything is there. But still, suppose, and this nucellus cells which are there, they are usually getting used up or getting degenerated. But over here they are still persisting. They are still persisting. Why? Because they also provide nourishment. So this persistent nucellus, my dear children, it is known as perisperm. It is known as perisperm. So usually it is degenerated, but still it's like too much love is there. So perisperm is there, still existing, and these are the cells which you get from nucellus. And you can see them only in few plants such as black pepper, both starts with BB, such as black pepper, and then you have beet. So black pepper and beet is the answer for this. So perisperm is the remnant of nucellus in some seeds after fertilization. If after fertilization, zygote and endosperm will be formed, right? Now it acts as a storage tissue for the developing embryo. Black pepper, that is, you don't have to remember this name, and beet is there. Okay, now the question comes, difference between perisperm and pericarp. What is pericarp? So listen to me carefully. So over here, when the ovule which is there, this is the ovule, inside the ovule zygote is being formed. Whenever we say fertilization happened and zygote has been formed, we will now call this ovule as a seed. We will now call this ovule as what? Seed. And this seed has the zygote, or basically it is going to form the embryo. So that's why when you put the seed in the soil, it will give rise to a whole plant because it has the embryo within it, the baby within it. Okay. Now over here, outside it, you have the ovary, my dear children. Outside it, you have the ovary. What happens to the ovary after fertilization? It becomes a fruit. It becomes a fruit. So there are different layers, walls of a fruit, my dear children. You call these walls of a fruit as pericarp. As what? Pericarp. It's not the outer layer I am talking about. All the layers in a fruit, my dear children, together it is known as pericarp. Okay. Pericarp. Please remember that. So it is the wall layers of the fruit, okay, after the formation, after the fertilization. Always write like that. So perisperm is the remnant of nucellus. Pericarp is the outer layer of, or basically the fruit walls that is formed post fertilization, after fertilization from the ovary. Done? Chalo. Now, why apple is known as false fruit? Which part of the flower forms the fruit? So, why apple is known as false fruit, my dear children? Basically, what happens, I told you, it's the ovary that becomes the fruit. But, suppose, over here, you know, flower rests on thalamus and then it is pedicel and everything is there. So, here is the flower and everything, and here you have the ovary. So, along with the ovary, okay, the thalamus which is there, it also thought that I also want to become the fruit. I am also going to help in the formation of a fruit. So over here, my dear children, what is happening? The fruit is being formed not only from the ovary which is there, but also from the other parts of the flower. These type of fruits are known as false fruits. Ma'am, apple can do this? Yes, apple can do this. So apple is actually made up of thalamus and also ovary. So don't mention ovary over there. You just say that apple is called false fruit because it develops not just from the ovary, but from the thalamus of the flower. But also from the thalamus of the flower, that will be much great. Or you can write, apple is known as false fruit because it develops from the thalamus as well. That's all. Easy. In apple, the thalamus enlarges and becomes fleshy, forming the edible part of the fruit. Okay, forming the edible part of the fruit. So true fruits are the one that developed only from the ovary, whereas false fruit developed from other floral parts along with the ovary. This definition has to be put first. Okay, so true fruits only develop from the ovary. False fruits are the ones that also develop from other floral parts, other parts of the flower along with the ovary. So apple is a false fruit. Why? Because it develops not just from the ovary. Please remember, not just from the ovary, but also from the thalamus of the flower. So ovary is still there. You will be having the ovary in the inside. Okay, the outer thing that you see, that is the thalamus. Okay, it has become enlarged and that is the thalamus. So here, please see, this particular picture is really important. You can be asked to label it as well. So you have the seed within, that means the ovule is there inside, and then you have the layers of ovary, my dear children, that is endocarp, mesocarp, and everything is a layer of the ovary. I told you when it becomes the fruit, it have different layers. Epicarp, endocarp and mesocarp are the three layers forming together the pericarp which is there. So all the things which are there inside, it is the ovary, and then outside which is there, it is the thalamus. Please remember that. Okay? Works? Now, How are parthenocarpic fruits produced by some plants and apomictic seeds by some other? First thing, what is parthenocarpy and what is apomixis? Listen to me carefully, okay? Okay, I told you that when fertilization happens, there is ovary. The ovary is the one that will be, so when the ovule over here is fertilized, it is now the seed, and the ovary which is there, it is now the fruit. Suppose I am telling you that without fertilization also, the ovary can turn into a fruit. Then the ovary is still turning into a fruit, but without fertilization. Then I'll call this fruit as parthenocarpic fruit. Ma'am, how can that happen? Basically, because of some hormones that is there in the plant. Because of some hormones which is there in the plant. So over here, listen to me carefully. I told that ovary is forming the fruit without fertilization. So without fertilization, do you think seed can form? Do you think seed can form? Over here, no. Over here, no. Okay, so in parthenocarpic fruit, since they are formed without fertilization, the ovule is now forming the seed. So can I say that they will be seedless? Can I say they will be seedless? Yes. So parthenocarpic fruits are the ones that are formed without fertilization from the ovary, and since fertilization has not happened, these variety of fruits will be what? Seedless. This is a natural process that occurs in the plants when there is some hormonal change occurring, but it can be induced as well. I can make plants do that as well. For example, I can make banana do that. Banana, you know, banana can be made like that. Banana also does not have much amount of seeds are there, but it's very small. Get it? I can make seedless grapes, I can make seedless watermelon out of this particular process if I induce certain hormones such as auxins. Get it? So parthenocarpic fruits are produced without fertilization of the ovary. They can be induced artificially using the growth hormones like auxins and gibberellin. Okay, example, you have banana, then you can make seedless grapes, seedless watermelon and everything else. Now you have apomictic seeds. What do you mean by that? That is kind of different. So over here, this was a scenario, and I am going to make the next scenario over here itself so that you can relate and understand. So my dear children, this is again the ovary. This is the ovule. Inside the ovule, I have the embryo sac. I am telling you, so until now you studied how is the female gamete being formed. Basically, megasporogenesis happens. The megaspore is there, one functional megaspore is there that will turn into embryo sac. And in the embryo sac, the cells will be formed like separately. I am telling you over here, the cells of the embryo sac, which should be haploid, which should be having half the number of chromosomes, they are not like that because of some other things that are happening in the plants, which is why they are all diploid. Which is why what? They are diploid. 2n. So the female gamete is already 2n. So does it need any n from the male gamete? It can still form the zygote. So this female gamete which is there, which is diploid in nature, or the embryo sac which is diploid in nature, basically what is happening over here, everything is diploid, which is why, okay, what will happen? This can directly form the zygote without fertilization. This can directly form the zygote without fertilization. And if zygote is being formed over here, can I call this as a seed? Can I call this as a seed? Yes, zygote is formed. So that means, yes, I can call it a seed as well. So this type of seed is known as apomictic seed. Now what is the importance of this, ma'am? You tell me. This female gamete is from parent. Suppose this particular plant parent which is having all the good features. Okay, I am reserving all the features just like that only. There is no fertilization with another male gamete or another male parent, my dear children. So whatever features I have in me is going to transfer to my progeny as well. Suppose I had good features, all the good features are going to get transmitted to my progeny. There is no chance of mixing of chromosomes which is there in sexual reproduction. So apomixis, this production process is known as apomixis, and this is a type of asexual reproduction. I call it, it's like asexual reproduction because I am going to make clones out of it. Clones as in same to same, exact copies I am going to make out of it. So apomictic seeds are really important. I am telling you again over here, fertilization was not happening, still fruit was being formed. Here also fertilization is not happening, still seed is also being formed. still seed is being formed. Why? Probably because the egg cell and everything were already diploid. They undergo all the division and all. So they don't need fertilization to happen over there. So apomictic seeds are produced without fertilization or meiosis. There is no meiosis happening. There is no reduction happening, my dear children. Reductional division is basically where 2n becomes n and n. Nothing of that sort is happening over here. Now they develop from the diploid cells like the nucellus or unfertilized egg. From the unfertilized egg that I told you, there is one more chance, that is nucellus. I told you there is ovule, and in the ovule the nucellus cells are there. These nucellus cells, my dear children, they also can get inside the embryo sac and basically give rise to what? Seeds. They can also give rise to multiple embryos, 1, 2, 3. There are multiple embryos, or basically multiple zygotes. When there anyway there is zygote, I'll call this thing as what? A seed now. But there's a condition that is being formed over here. So cells of the nucellus are getting inside the what? Inside the embryo sac. So, and so many of them are going to form, get inside, and so many embryos are going to form. This is known as polyembryony. This is known as polyembryony. So this is also a sub effect of what? Apomixis. Please not mix all of this, okay? Apomixis can happen if from the unfertilized egg or from the cells of the nucleus also. Anyways, they are going to form the zygote or the embryo. Now this condition, second condition that I explained, is an example of polyembryony as well, where multiple embryos are being formed within the same seed. Okay, within the same seed, multiple embryos are being formed. This is found in citrus and mango. This is found in citrus and mango. What about the unfertilized egg and everything? This happens in some grasses and also, some grasses and things that belongs to Asteraceae family. So citrus and mango is an example of apomixis, but is also an example of polyembryony. They make multiple embryos with the help of the cells of the nucellus. Okay, so I've explained three things over here. One is parthenocarpic, the next one is apomictic seeds, then I explained about polyembryony, which is also a subdivision of apomixis. I hope I am making you understand things, you're not getting confused, because I really don't want you to get confused, okay? Now going ahead, define polyembryony, mention the plant which exhibits polyembryony. So polyembryony is the occurrence of more than one embryo in a seed. I told you it can arise due to formation of embryos from nucellar or some other cells, but not a fertilized zygote. This is not a fertilized zygote. Basically it is due to the formation of the embryos from the nucellar cells which are there. Example, citrus fruits such as orange, lemon, etc. They exhibit polyembryony. Works? Now, just a second. So there is a question related to this only. Just let me see that. I have explained it somewhere and I want to explain it now to you. Banana is a parthenocarpic fruit, whereas oranges show polyembryony. How are they different from each other with respect to seeds? Banana is formed from parthenocarpic fruit. Do they form parthenocarpic fruit? That means fertilization has not happened. The ovary has become the fruit by itself. There is no fertilization. Will there be seed? There is no seed. Okay. What about oranges that show polyembryony? Oranges, they show polyembryony, that means they are showing apomixis over here, my dear children, where what is happening? Without fertilization, still seed is being formed for sure. But this seed can have multiple embryos or single embryo, whatever is there. If multiple embryos are there, it comes from the nucleus cells. So what is the seed? Seed is there or not? Seed is there and seed will be having multiple embryos as well. Okay, now how do you explain the answer? You can explain it like this. Banana is a parthenocarpic fruit, orange is a result of polyembryony. Okay, seed formation does not produce seed, they're seedless fruits, but here they will be producing what? Embryos, multiple embryos in one seed. Now reason, it is formed without fertilization, parthenocarpy, here occurs due to presence of extra embryos from these nuclear or synergid cells. You just have to mention nuclear cells, you don't have to mention synergid because they are not there in your NCERT. So it occurs due to what? Presence of extra embryos from the what? Nuclear cells. Over here it is directly just a result of what? Fertilization, or probably you can write because of some hormones. Because of hormones, growth hormones like auxin fertilization, it is formed without fertilization. That is better. So reason is this. Because of certain growth hormones, what is happening? The ovary is getting converted to fruit without fertilization. Over here it occurs due to presence of extra embryos from the nuclear cells. It is naturally, example, naturally seedless seeds contain more. Okay, you don't have to write this point. So I hope that you will write all these 3 points, and now let's go back to where we were. Where were we? We were here. Okay. Question number 12. Why are some seeds referred to as apomictic seeds? Mention its advantages to a farmer who uses them. So you already know the definition. Apomictic seeds are those produced without fertilization, meaning they develop from diploid cells of the ovule bypassing the process of meiosis and syngamy. That means bypassing the process of meiosis, as in there is no meiosis happening, and also syngamy. Syngamy means fusion of male and the female gametes. That is what you mean by syngamy. That process is known as syngamy, male and the female gamete. Okay, so bypassing all that, you're directly forming the seed from the diploid cells of the ovule. Now advantages, you tell me the advantages. Children, please don't look over here. Okay, so what are the advantages? I told you it's like exact clone. So for example, I have some plants. Okay, for example, the farmer is there. So farmer thinks that this plant is so good, yeah, this is pest resistant, this is that, this is this. But basically, if I let it undergo fertilization like that, what will happen? There are chances that it can, mix and merge. There are other features that will be coming. I do not want that. So these apomictic seeds, which are basically from one particular sex, that is the female plant only, and if the plant is having all the good features, why can't I just use those seeds to produce more of them? So that is one thing. So genetic uniformity, since apomictic seeds are clones of the parent plant, they ensure uniformity in the crop characters. All the crops will be having the uniform character, my dear children. The genetic uniformity is there. Cost effective. Farmers do not need to buy hybrid seeds. Do they need to buy hybrid seeds? Because the hybrid seeds have good quality, everything. So do they need to buy that? No, these apomictic plants, my dear children, they are going to produce apomictic seeds only, right? So obviously, it's over and over generation you will be having the same type of things. So farmers do not need to buy hybrid seeds every season, as apomictic seeds maintain desirable traits over generation. So it is cost effective, very important point, and its genetic uniformity is also very important. Okay, now I think you understand things better, right? Now write any two commercial uses of pollen grains and mention the viability period of pollen grains of Solanaceae family. Solanaceae family as in the one where potato and everything comes. You like potato? I like potato. I love potato. Okay. So you're asked about the commercial use of pollen grains. So commercial use of pollen grains, one is that basically it is acting as a nutrient supplement. You know, sportsmen also who run and do exercise like this also. So all of them, they rely on pollen grains as well. You might not believe it, but yes, since pollen grains are there, you know, vegetative cell and generative cell is there. The generative cell is the one that forms the gametes. The vegetative cell provides nourishment to the gametes. So if this nourishing thing is there in the pollen grains, why can't we take it? Yes, we can also take it. So pollen grains are rich in nutrients and are used in health supplements to enhance what? Stamina and immunity. What about other thing? The next thing is pollen banks. What is the commercial use of pollen banks? If these pollen grains are stored, my dear children, I can basically use this pollen grains whenever I need to make a new crop. Right, now the crop improvement program, if I have good quality pollen grains from good quality plants, my dear children, I can just take it and use in the crop improvement programs. So pollen grains are stored in liquid nitrogen, that is in minus 196 degrees Celsius. Liquid nitrogen and also in minus 196 degrees Celsius. Please remember this, okay? And they can be used for artificial hybridization. I can artificially hybridize it. I can take the pollen grains, put on the stigma of the flower which I want, the plant which I want, and then basically desirable characters are coming together. Now the crop is totally improved. So pollen banks and nutritional supplements is one thing. Now you had asked about the viability period. What do you mean by viability? Viability means the ability of the pollen grains to germinate and basically fertilize. So the viability of pollen grains of Solanaceae family, what about that? Is it viable for few months or few seconds or things like that? So pollen grains of Solanaceae family remain viable for months under suitable condition. This is one good thing about them. So in your book, other examples are also given of rice, cereals such as rice and wheat. You know how fast they lose their viability? Within 30 minutes. Rice and wheat that we eat, my dear children, they lose their viability within 30 minutes. So if they are not landing on the stigma and probably germinating, and the viability will be lost within those 30 minutes. So only 30 minutes. Here it is viable for months. This is very much important. Please remember it. Again, an MCQ question can be coming out of it. Okay, now what is artificial hybridization? Name the techniques that are employed to achieve this. Artificial hybridization, as in I am artificially going to do something, and hybridization, that means I am going to make two things come together. Okay, so over here what do I do is, suppose I have a very beautiful pretty little flower, okay, like you guys, pretty little flowers, okay. Now it is a controlled reproductive process. Why do I say controlled? Because it is under my control, that is human's control. Controlled reproductive process in which desired pollens from one plant is manually transferred to the stigma of another plant to obtain improved hybrid varieties. So what do I do is, I have a plant over here with the flowers and the pollen grains and everything, but I don't want the pollen grains to land over here. Okay, I don't want this. I'll take another plant. Okay, this was not you. Okay, this was not you because you guys are so good. Okay, this is you. So from here, this is another flower, my dear children. From here, what I'm going to do is, I'm going to collect some pollen grains. I'm going to collect some pollen grains and put it on the stigma over here because I want to make a new thing that is up there. But the problem is, this guy already had the pollen grains. It can also land. That's the problem, right? So how do you achieve this? First thing, emasculation. Masculation as in masculine, as in male. Emasculation as in I'm removing the male thing. So removal of anthers from the bisexual flowers. So if the flowers are bisexual, that means it has two sexes over here. I am going to remove what? Remove the male part from this. Tada! This is known as emasculation. Why do I do it? Because I want to prevent the pollen grains coming and landing over here. Second thing, there are other flowers also. From here also the pollen grains can come and land over here. I don't want that. So what do? I have to make it safe really fast. So I'm going to bag it. I'm going to cover this. So covering the emasculated flower with a bag to prevent what? Unwanted pollen contamination. I don't want that. Now, when the stigma is receptive, I know that. What do? I take the pollen grains from the plant that I desire and I'm going to put it, I'm going to open this bag and I'm going to put this pollen grains on it. And then I'm going to do something that is known as rebagging. I have rebagged. So desired pollen is transferred to the stigma and the flower is what? Rebagged to ensure fertilization. Fertilization will happen now. You're getting what? Hybrid plant that is going to come out there. This is known as artificial hybridization. Is it occurring naturally? It is controlled reproductive process, my dear children. But desired pollens from one plant is manually as in we are doing it, okay, manually transferred to the stigma of another plant to produce hybrid varieties. Works. This is artificial hybridization. These are the steps. Now, name the product of fertilization that forms the kernel of coconut. How does the kernel differ from the coconut water? From here, I have to tell you one very important thing. I told you that there is a male gamete that fuses with the female gamete, didn't I? Okay, it produces zygote. It produces zygote. What do you call this process as? This process is known as syngamy. I told you this is known as syngamy. This is the first thing. But there are two male gametes. The other male gamete fuses with what? The two polar nuclei of the central cell. And now you have N and N and N. You got 3N. What are you forming over here? You are forming endosperm. What do you call this fusion as? You call this as triple fusion. Why triple fusion? 3 nucleus are coming over here. 1 nucleus of the male gamete, 2 nucleus of the polar nuclei. 3 nucleus are coming over here, which is why I call it triple fusion. So syngamy is happening where zygote is formed. Triple fusion is happening where endosperm is formed together. Since there are 2 fertilization happening, I'll call this as double fertilization. I'll call this as double fertilization, my dear children. Okay, one very important thing over here is that this is the primary endosperm nucleus, primary endosperm nucleus that is being formed after the nucleus are fusing. The cell which is there, you call it primary endosperm cell, which is being formed. Now this primary endosperm cell has to make sure that it is dividing continuously to basically nourish the zygote to form the embryo. When it divides, the nucleus keeps on dividing. The nucleus keeps on dividing, but the cell does not. So usually the nucleus keeps on dividing. Later there will be cell wall formation. But in some, still the nucleus will be free. So this is free nuclear endosperm. This is what basically the cellular endosperm which is there. This is what? The cellular endosperm which is now like a cell. Whereas there are free nuclear endosperm which has not been covered by any cell wall. So in the case of the coconut that you see, the coconut water contains the free nuclear endosperm. They are not covered by anything. The kernel, which is the white color thing that you eat, my dear children, it is cellular endosperm. So aren't you eating the food of the embryo? Yes, you are. So later if it's kept like that only, if you might have seen, okay, inside that the embryo will be forming with the help of this endosperm which is there. But we do not keep like that. We just take it and drink it. Okay, so the product of fertilization that forms the kernel of the coconut is the endosperm. What forms the product of fertilization? which product? You have two products. One is zygote, the other one is endosperm. So which product is there that helps in the formation of the kernel of the coconut? It is endosperm, my dear children. How does the kernel different from the coconut water? I told you, the endosperm which is there, my dear children, it can be the cells, the nucleus which is there. The nucleus keeps on dividing. Some get surrounded by the cell wall and everything, some does not. That does not is known as coconut water. It is a free nuclear liquid endosperm which is rich in nutrients and provides nourishment to the developing embryo whenever it develops. Now kernel which is there, it is a cellular endosperm that gradually solidifies as the coconut matures. Now uses of endosperm is what? It provides nutrition to the developing embryo essential for the seedling's growth. Now you might have seen in some coconut there is no water because all of it has been utilized for the formation of the kernel, the cellular endosperm, which is further nourishing, going to nourish what? The embryo. Works? Understood? So next time you eat the coconut, please remember you're eating endosperm. Now explain any three advantages the seeds offer to angiosperms. Again, these if you have seen the questions which are there, almost few questions, okay, few questions might be common ones which are there in the model question paper and everything. Few questions are something that is not given priority to in the model question papers, my dear children, which is why I have included them over here. Okay, so make sure you're reading. This is from the question bank. Any question can pop up, and if you think that this is important, this is not, you might lose your marks. Okay, so advantages of seed to angiosperm is what? Whenever seed is there, seed is having the layers, my dear children. The seed will be having the protective layers. Do you know that? The seed has seed coat. The seed which is there, the ovule which is there, it also has seed coat. It's not just the ovary that has all the layers. So seed coat is made up of tegmen and oh my god, testa and tegmen. Yeah, testa and tegmen. Okay, those are the coverings of the seed. Now, when the seeds are covered, obviously it will let the seed survive during unfavorable condition as well. And if it is surviving under unfavorable condition, basically it's really good, right? It helps in survival. Later on, when favorable condition come, it can germinate. So better survival and protection. Seeds are enclosed in a protective seed coat which safeguards the embryo from mechanical damage. So what is the use of seeds to the angiosperms? Basically, their little embryos are being provided protection with the help of this. Okay, so it is anything against mechanical damage, desiccation and unfavorable condition, it can still survive because it has a seed coat. Second thing, efficient dispersal and colonization. Seeds have specialized structures such as wings, hooks or fleshy coverings, my dear children, that aid in dispersal by wind, water, animals, helping plants spread to new areas. So obviously for survival of the plants, my dear children, to make their species continue, you need dispersal to happen. Yeah, it has to be dispersed. So because seeds are there and seeds have all these cute little features, it lets the plant survive. It lets continuity of the species. You can write like that. So efficient dispersal and colonization. They come somewhere and they grow somewhere. That is colonization. You get it? Now dormancy and germination control. Seeds can remain. Suppose favorable condition is not there. You spoke about protection and everything. Okay, it is protected and everything. Whenever favorable condition is there, it arises. But it should also have the knowledge that I do not have to germinate under unfavorable conditions, right? So seeds basically can remain dormant. And for having that feature, it has to remain dormant. Sorry, I just meant if it has to basically sustain through unfavorable condition, it should be having the property of remaining dormant as well. So seeds have that. Seeds can remain dormant until favorable conditions arise, ensuring survival of the species during harsh environments as well. So better survival, this points might be mixed, but you can put in 2 points and explain, because that's how we can do. Okay, so better survival and protection, efficient dispersal and colonization, and dormancy and germination control. Works. Next thing, answer the following. Explain different types of pollination based on the source of the pollen. List the characteristic features of water pollinated flowers. So I told you already there are 3 types of pollination: autogamy, xenogamy and geitonogamy. I'm not going to go beyond that. This question is important. So autogamy is basically if a flower is there, the pollen grains and the male and the female part is there in the same flower. From here to here, if it's happened, it is known as autogamy. Now if different flowers but of the same plant, my dear children, from here to here, if it is happening, it is known as geitonogamy. 2 different plants and 2 different flowers over here to here it's happening, it's known as xenogamy. Now list the characteristic feature of water pollinated flowers. That is important. So how do you write it? First thing, you can go through the answer if you want. The PDF will be provided to you in the WhatsApp channel, so please do not worry. Okay, so pollen from the anther is transferred to the stigma of the same flower. Example, pea, rice and things like that. You don't have to remember the examples if it's too cumbersome for you. Okay, I won't recommend that. But you should know the monoecious plants and dioecious plant example. Okay, now characteristics features of water pollinated flowers. What do you think? they should be light and they should be unwettable pollen. If they are getting wet, my dear children, basically they will be sinking down. So pollen grains are light and covered with a mucilaginous coating to prevent what? Wetting. Second thing, long and thread-like stigma should be there. Stigma is the one that receives it, right? So pollen grains, if they are light and covered with mucilaginous covering, the other thing that the stigma should be having is that it should be feathery, it should be or else ribbon-like to capture pollen from the water currents. You get it? Stigma should be feathery or ribbon-like to capture pollens. Now, no fragrance or nectar. Water pollinated flowers do not produce any scent or nectar to attract the pollinators. Filamentous pollen release. Some plants release pollen grains in long filaments for easy dispersal in water. Okay, and some have basically ribbon-like pollen grains as well. Just let me check. Okay, once. I think there's a confusion over here. Okay, the confusion is sorted over here. Listen to me carefully. Okay. The thing is, where was I? So over here, instead of stigmas being feathery and things like that, you can just write about the pollen grains. You can just write about the pollen grains. That will be way better. So you can write that the pollen release which is there, filamentous pollen release, or basically different shapes of pollen release, okay, or of pollen. That is, some are ribbon-like as well, some are filamentous. Since they are ribbon-like, it will help in the movement through the water currents. That will be better. So do not please write this, it will be bad. Okay, so instead of that, you can write the stigma and the style which is there. Style is long. Style is long for receiving pollen grains. Usually this applies, you know what, in the water pollinated flowers, there are two types of flowers. One that basically does the pollination at the surface level itself. So pollen grains are released on the surface, and the style from the down, it will be coming upwards just to receive the pollen grains up over here, my dear children. So in this case, the style is very long. On the other hand, there are certain plants where basically the pollen grains are released within the water itself, in the submerged state as well, such as in the sea grasses. In the sea grasses, it is in the Zostera and all, it is in the submerged state. Whereas when you talk about Vallisneria, it is in the surface level. You get it? So there are different features. So light and unwettable pollen, this is something unique, very similar for both of them. Now style is long for receiving pollen grains. This is found in Vallisneria, my dear children. Vallisneria, where pollination is happening in the surface level. So you can write where surface level pollination happens with the help of water, there the style would be long for receiving the pollen grains. Example in Vallisneria. Now you can write the next thing. For example, in the plants which are submerged, okay, the pollen grains would be ribbon-like so that they could be easily moving through with the help of the water current such as Zostera. Okay. Can I write that? And this point is really important. No fragrance will be nectar will be there because there is no pollinators. There is no need to attract the pollinators. It's just the water current. So please write like that. Examples you can mix up with this and write the points. Okay. Again, I am saying this is not given in your model question paper, but this is there in the QP. Okay, the question bank which is there. So until now in the model question paper, insect pollinated flowers features and the wind pollinated flower features are being asked, but not water pollinated. So I am explaining water pollinated as well because what if it comes? You get it? So just for your understanding, but please give priority to wind and insect first, especially insect. Now, going ahead, pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition. Explain. So what do you mean by that? Pollen-pistil interaction, as in pollen is a male gametophyte. It is going to land on the pistil. This whole structure which is there, that is the ovary, style and the stigma, this is known as the pistil. Whole thing is known as the pistil. So the pollen grains will come and land over here. Now, there are two decisions that has to be made. One, do we need to allow pollen germination, that is pollen tube, can it be formed or not? Second, should we inhibit it? So pollen-pistil interaction is actually a very dynamic process and there are lots of exchange of lot of information between this. You get it? Which allows it to promote or inhibit. So we have to explain that. So how do we explain? So first thing, recognition of pollen. The pollen recognizes the pollen landing on its stigma through chemical signals. The pistil recognizes through chemical signals. It can distinguish between compatible, that means of the same species, and incompatible, that is not different species, pollen. Now, promotion of compatible pollen. What if it is compatible then? If the pollen is compatible, the stigma secretes substances that helps in the pollen hydration and germination. The stigma itself will be releasing something that will allow the pollen grain which is there to germinate and for the formation of what? Tube. And this tube will go through this style, my dear children. The pollen tube grows to the style towards the ovule for fertilization, and it will be carrying the male gametes. Now, on the other hand, inhibition of incompatible pollen. If the pollen is incompatible, the pistil will prevent what? Germination or pollen tube growth. This prevents self-sterilization or self-incompatibility study, right? And ensures what? Cross-pollination to happen. Now this, and ensures, well, basically you do not have to write this. Works. Now, role of chemical signals. Various chemical compounds regulate pollen tube growth and guide it towards the ovule. These interactions ensure successful fertilization in compatible cases. Significance: ensures genetic diversity in angiosperms, prevents inbreeding depression by rejecting self-pollen in self-incompatible plant as well. So it ensures genetic diversity, as in it allows the other plants also to come over here and fertilize. And the second thing, sometimes our own pollen grains are incompatible. In that way also we are basically increasing the genetic diversity, right? We are not allowing our own pollen grains, but we are making others to come and fertilize over here. So it is going to increase the genetic diversity. We are making new type of plants. It is helping in cross-pollination rather than self-pollination. Okay, so these are the significance that you can write if you want to, but this is the whole thing. The question says this, you just have to explain the process of inhibition and compatibility, but you can write extra things if needed, if it's needed to support your answer. Now, answer the following. Plan an experiment and prepare a flowchart of steps that you would follow to ensure that the seeds are formed only from the desired sets of pollen grains. So, here, seeds are formed only from the desired set. Whose desired set? Our desired set. So, what is this process? You're talking about artificial hybridization. So, you have to talk about the steps, emasculation, then bagging, okay? Then bagging, and then pollination and rebagging, all this thing you have to explain. Name the type of experiment that you carried out, my dear children. It is artificial hybridization. Write the importance of certain experiments, that you have to write it. So I told you, selection of parent plants, emasculation, bagging, pollination, rebagging, and then harvesting, and everything is there. Okay. Now if you talk about the importance, helps in crop improvement by producing hybrids with desirable traits such as what? Disease resistance, high yield, and everything. Ensures genetic purity of crops in plant breeding programs and prevents unwanted cross-pollination and maintains specific plant characteristics. We do not want unwanted cross-pollination, or basically we do not want inbreeding depression also. That is also one point. We can prevent inbreeding depression by doing this. Get it? We did not allow self-pollination to happen. We ensure that cross-pollination is happening. And we also ensure that unwanted cross-pollination is not happening over here. We don't want bad plants to come and fuse and give a bad progeny. You get it? So that is one thing. So will you be able to write it? I hope you do. Now answer the following. Double fertilization is reported in plants of both castor and groundnut. However, mature seed of groundnut is non-albuminous and castor is albuminous. Explain the post-fertilization events that are responsible for it. What? So, double fertilization, I told you, triple fusion as well as syngamy. It is reported in the plants of castor and groundnut. There are two plants, castor and groundnut. Okay. Now, however, the mature seed of groundnut is non-albuminous. What do we mean by that? Groundnut is non-albuminous. The seed is non-albuminous. That means it is dicot. Non-albuminous means the endosperm is totally utilized. Non-albuminous means there is no persistent endosperm. The endosperm is completely utilized. And what are they forming out of it? Cotyledons. Okay. Now, in the case of castor, so groundnut, as you said, it can be broken into two, my dear children. So, remember, it is non-albuminous. It is non-albuminous because no persistent endosperm is there, since we have already made cotyledons out of it. Whereas, on the other hand, castor, maize and everything, it is albuminous. Why it is albuminous? There is little amount of endosperm. I told you monocots are really good. They leave little amount of endosperm and they just make one cotyledon, monocot. There is only one cotyledon. So endosperm is still persisting, which is why it is known as what? Albuminous. Explain the post-fertilization events that are responsible for this. What is the post-fertilization event that is responsible for this change, my dear children? So double fertilization occurs in both castor and groundnut, leading to this. Now in castor, the endosperm is not completely consumed by the developing embryo. This is the post-fertilization thing. After fertilization, zygote is formed, and from zygote, embryo is being formed. So what is the change over here? The endosperm is not consumed by the developing embryo completely. The mature seed retains a significant amount of endosperm, making it what? Albuminous. This is the post-fertilization chain that leads to this. Now, in groundnut, the endosperm is completely consumed by the embryo during the development. As a result, the mature would lack the endosperm, making it non-albuminous. So, don't have to mention about cotyledons also, because you're talking about albuminous or non-albuminous. Okay, then you can, if you want, you can say that yes. Instead, it forms 2 cotyledons for nourishment. That's all, if you need. But you do not need it. You just say it's completely utilized over there. We do not say where it is completely utilized, or no, you do not have to give the explanation for that. Over here, you say it's completely utilized. My dear children, I know that I might be explaining in detail, but trust me, it is going to be really important. In this way, we are covering the whole chapter as well. Okay, so please do not judge me on the basis of that. This is for your own good. So now there are little diagrams, my dear children. The questions have been done. The theoretical questions have been done. Please make sure you are practicing the insect pollinated, the characters of the insect pollinated flowers. I can tell you a few. Okay, they will be rich in nectar, or basically some rewards. They will be giving rewards. The flowers which are there, they will be coming together to form an inflorescence, so that they appear to be big and things like that. They would be fragrant. They would be nice because you have to attract the pollinators, and also the pollen grains will be sticky. The pollen grains, opposite of wind pollinated. Okay, over here it will be sticky. Why? Because you want the pollinators to have their on their body, you want the pollens to be stick, so that it can be easily moving around, right? So that's why. And yeah, I think that sums up. At least these many points should be written down. You go and read by yourself also. This is a very, very important thing. Okay, now draw the transverse section of young anther. Very important. So young anther, my dear children, it is bilobed and it is dithecous. So bilobed as in it has 2 lobes, and it is dithecous as in you have one tooth theca over here. It is dithecous. Now inside each lobe, my dear children, you might be seeing this. This is sporogenous tissue. Okay, inside this there are so many cells which will be giving rise to the microspores. Outside this, just outside the sporogenous tissue, there is a layer. This is a very important question. Okay, there is a layer which provides nourishment to the sporogenous tissue cells. What is this layer known as? This is known as tapetum, my dear children. What do you call this? Tapetum. What does tapetum do? Tapetum provides nourishment. And please remember the feature of tapetum. It is not uninucleate. It does not have just one nucleus. It can have more than one nucleus, and provides nourishment to this sporogenous tissue. Outside this, if you talk about the outer layer, starting from the outside layer, you have epidermis first. Okay, then you have endothecium. You have epidermis, then endothecium. Then inside that you have middle layer, endothecium and the tapetum. In between that you have a middle layer also. So epidermis, endothecium, middle layer. Okay, epidermis, endothecium, middle layer, and tapetum, and then sporogenous tissue. This is the thing that we have to follow. Please don't write endothecium outside. Epidermis is also always going to be outside. So epi, endo, inside, endo as in inside, epidermis, endothecium, middle layer, tapetum, and this is a, what do you call it, a MCQ question as well. Please make your diagram just like this. It's very, 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 very important. Okay, you can zoom in and show there are cells, or you can just show the zoomed in version where there are cells like this. Not like this. Okay, make it properly. And then there is tapetum, again made up of cells. Make sure it is rectangular cells that has been mentioned over here, or polygonal cells shape that is being mentioned over here. Okay, and go like that. Works. Not gonna go in detail of the videos. It's a very important thing. Draw a LS, okay, lateral section of grass embryo, and label the following parts. What is a grass embryo? Grass embryo has it talking about monocots. Grass is a monocot, my dear children. So you have to mention scutellum, coleoptile, radicle, root cap, shoot apex, coleoryza. Listen, I'll explain your basic funda. Okay, listen to me carefully. I told you that there is an embryonal axis which will be giving rise to what? Root and the shoot. Root and the shoot. Now over here, this is the embryonal axis. Okay. This end is known as plumule, which is the shoot apex. This is known as radicle, which is the root apex. It gives rise to the root, this gives rise to the shoot. Now radicle is covered by root cap. Okay, and over here one endosperm is needed because it is a monocot. So this is the endosperm that we have. This endosperm has a, sorry, not endosperm, cotyledon. Okay, and this one cotyledon is needed. So this is a cotyledon, and this cotyledon's name is scutellum. Do you call it? Scutellum. I told you in monocots the endosperm is still persisting, so you have little amount of endosperm as well. Okay, the little amount of endosperm is also there. Now the plumule is actually covered by something which is known as coleoptile. which is known as coleoptile, which gives rise to the leaves and everything up there. And over here also it is covered by something that is known as coleorhiza. Coleorhiza. So this is the entire thing which has been mentioned over here in a much complicated way, but we have to remember it. So listen, you have to practice and draw. This is very, very, very important diagrams. All the first diagrams I am showing you are the important ones, okay, the most important ones. So you see shoot apex which is there, it is covered by coleoptile, okay, and over here you have root radicle, then root cap, then coleorhiza, just like how I mentioned. Over here you have scutellum, one cotyledon, my dear children, endosperm is there, not been mentioned over here, that's the only thing. Okay, make it properly, draw it. I know it looks like very bizarre drawing, but yes. Okay, I cannot go ahead with helping you how to draw this. This will be taking too much of your time. So please use your limited knowledge and try to draw it. Okay, next thing, draw a neatly labeled diagram of typical anatropous ovule. What do you mean by that? So first thing first, so listen to me. If I show you normal ovule, you will be able to understand anatropous ovule as well. So over here in the normal ovule, suppose this is the ovary. In the ovary there is the ovule. Listen to me carefully. In the ovarian cavity, there is something that is known as placenta, which is going to provide nourishment to the ovule. It is linked to the ovule with the help of something that is known as funicle. What? Funicle. Funicle attaches at a place, this is known as hilum. The ovule which is there, it has two outer layers, okay, or basically layers. This is inner integument and this is outer integument, okay. Outer and inner integument, fine. And this opening which is there, it is known as micropyle, and this end which is there, it is known as micropylar end. Micropylar end, and this is where the embryo sac is there. Remember, in the embryo sac, wherever the egg cell is there, that is the micropylar end, and opposite to this end it will be chalazial end. Agreed? And inside that there will be nucleus and everything, and the nucleus cell one will be dividing to form the embryo sac. Listen to me carefully. Suppose the placenta and the funicle is shifted somewhere here. It is shifted somewhere here. Where is the chalazial end now? Chalazial end will be still here because micropylar is going to be here itself. You get it? So this is anatropous ovule, where what is happening? The placenta and funicle which has to be here, it is now here. So funicle is here, placenta is not being made, and the place where it attaches, it is known as hilum. And over here you have the micropyle and everything. Chalazial end is still at the opposite side. Got it? Okay. Inside that, please mention nucleus. You can mention the cells of the nucleus and then make an embryo sac also within it. Done. Done. This is anatropous ovule. Okay. Now, in your book, wherever anatropous ovule is there, there is example of syncarpus and apocarpus, carpillary system and all. Just go through the examples of that. It's very, very important. Syncarpus example. Syncarpus means the carpels which are there, the pistils which are there. There are so many, but they are all of them are fused. Example is papaware. Okay. Now, apocarpus as in the carpels are there, but they are different from each other. They are separated. Example is Michaelia. Just go through that picture once. Okay. So this is very important. If you talk about dicots, my dear children, dicots is even more simple. You have the embryonal axis. This is plumule, this is radicle, and there is root cap. Okay. Now, in them there are two cotyledons. So there is one cotyledon and there is another cotyledon. So inside your book, in your book, no, they have mentioned it something like this. Listen. So made cotyledon like this. So these are the two cotyledons. This is the plumule region that you have the radical region. Okay, below the embryonal axis, my dear children, this is known as hypocotyl. Above the embryonal axis, whatever is there, it is known as epicotyl. And in the epicotyl, you will be having the plumule. In the hypocotyl, you will be having the radical. This is dicot embryo. Make it properly, you will get marks. Label it properly. Please label with a pencil, that will be much better. Please draw with a pencil. You can label with pen also, but that is not something that I would recommend. Okay, now draw a neat label diagram of mature embryo sac. How do you make it? Mature embryo sac, what happens? The functional megaspore is there, right? The nucleus, one comes, okay, it is now going to undergo division, first mitotic division. So nucleus first divides into two nucleus. This nucleus also divides into two nucleus. Here also, two nucleus. This also divides again into, so 1, 2. So how many division? First mitotic division of nucleus, second, and then the third. At the end, 1, 2, 3, 4, 5, 6, 7, 8 nucleus are there. How do I arrange it? So one nucleus. Each of them are in one cell. These are antipodal cells. Okay. Now over here you have two cells, These are the synergid cells. And this cell is the egg cell. 1, 2, 3, 4, 5, 6 nucleus I have divided. What about the 2 nucleus? 2 nucleus, I am just going to keep it over here. This is the 2 polar nuclei. This is the 2 polar nuclei, and this is the part of the central cell. Please mention this is the micropolar end and this is the chalazial end, since the egg cell is over here. Make the filiform apparatus also. Done. You're done with the embryo sac. Done? Done. Now, draw a neatly labeled diagram of monocot seed. That was embryo, this is seed. In seed, what do you do? In seed, what do you do? Basically, you mention all this coleoptile, plumule, radicle and everything, and the scutellum, and then you mention about the endosperm as well. Please mention the endosperm, okay? And outside that, there will be pericarp. Actually, you're talking about the, okay. You can mention the pericarp, which is the outer layer of the fruit. So, in some fruit, what is happening is that they have fleshy layers. So, pericarp will be differentiated into different layers. In some, there is no fleshy layer, so pericarp will be just the outer layer which is there. So, please mention the endosperm, pericarp and scutellum and everything up over here. This is a monocot seed, not an embryo. Embryo is different. Okay? Seed, inside the seed you have the embryo, right? So, this is the whole seed. How does it look? Now, that's all. I'll request you. It's almost like 1 hour 37 minutes. Okay, I'll request you to take a break. Okay, then come back. From now on, we are going to deal it really fast. Okay, we are going to pace up a bit because otherwise it might not get completed. Okay, so amount of explanation that I am giving might be way too much, but I know it is helping you. But children, since we have less time and you have to revise all of this thing at night, I do not want you to waste any bit of your time. So let's take a break over here. Let's come back within 5 minutes. Okay, within 5 minutes, I'm also gonna take a break. You also take a break. Come back within 5 minutes. You're not going anywhere. We have to study the 4 chapters today itself. 1 chapter is done. Give a big round of applause and tap your back. Hello my champions, I hope you're back from your 5-minute break and you are here physically, mentally, emotionally, structurally, everything. Well, from now on, as I said, we'll pace it up a bit because I don't want you to say that, Jasmine ma'am, she taught for 3 to 4 hours after the English exam. She does not know that we want to study or what. Sorry, we want to sleep or what. So, yes, I definitely know that you want to sleep, my dear children. But I was also making sure that you are understanding things as well. I don't want to pressurize you with just questions and not no explanation, you get it. But I do get it that you might be tired and everything, and this went really long, so I don't want this particular video to go so long. I just want it to be useful for you, so I will pace it up a bit for the children who basically know certain things, so there is no need for them to revise a lot. But I definitely understand there are children who, as I said, might not have gone through the chapter really well. So for them, I'll maintain the pace. If you feel the pace is way too much, what you can do is there is a setting, button over there. You can control the speed over there. You can make it less. Okay. So I am giving the remote control to you. You can manage me however you want. So it's just like adding salt. Some people like salt. Some people do not like salt. So according to your own will, you can basically control my speed. Right? Can we get started? I hope we can. Let's begin with human reproduction, which carries 11 or 10 marks. So according to one blueprint, it is 11 marks. According to the next blueprint, it is 10 marks, which is why it is given like that. The first question over here, my dear children, is list the accessory ducts of male reproductive system in humans. What do you mean by accessory ducts? The ones that will be carrying the sperms. Where are the sperms made in the case of human males? It is made in the testis. It is made in the testis. Now from the testis, it has to be carried towards the urethra, because that is how it will be removed out from the body, right? So who will carry it? Accessory ducts, my dear children. So please remember, if we talk about the testis, each testis, my dear children, will be having testicular lobules. What? Testicular lobules. So this shape, this septa that you see over here, so they are testicular lobules, my dear children, and inside each of them you will be finding 1 to 3 highly called seminiferous tubules. So there are so many seminiferous tubules as well, and seminiferous tubules is the place where the sperms are being made. From the seminiferous tubule, suppose this is a tubule, testicular lobule. This is also a testicular lobule. Here is the seminiferous tubule, and from here it will be moving out. Okay, it will be moving out, making a structure like this, which is known as rated testis. That is the first thing that we have according to your NCERT. Now the rated testis, again, this is happening inside the testis. Okay, rated testis again will be forming another duct over here, which is known as vasa efferentia. Okay, vasa efferentia. Now, finally, it will be moving out of the testis and would be coiling around here on one side of the testis, my dear children. this particular structure that you see over here is epididymis. What do you call it? Epididymis. It is almost 6 meters long, but it is coiled. Okay. Now from epididymis, the sperms are being carried towards our body. So basically testes are outside the abdominal cavity. Now vas deferens would be carrying the sperms towards inside, towards the urinary bladder and then to the urethra. So basically it goes until what? The urinary bladder. The vas deferens would be going until the urinary bladder. So this is the next thing. Now when the vas deferens basically reaches, suppose this is a urinary bladder that we have over here. It is opening into the urethra. Okay. Now we have our vas deferens over here from both the sides. Now vas deferens will be receiving a secretion from a accessory gland. Remember, it's not a duct, it's a gland. Now this gland is going to give out some secretion to the sperms which are being carried through the vas deferens. Now together they are going to open over here. Together both of them are going to open here with the help of an ejaculatory duct. With the help of an ejaculatory duct. Now ejaculatory duct will be opening into something that is known as urethra. So ejaculatory duct, and then we have what? Urethra, through which that will be finally moving out. So these ducts help in the transport and storage and maturation of sperms before they are exited from the body. Cool. So you can write after that, you can write urethra, and that's all. Okay. Urethra is the last one that we have. It ends, the opening of the urethra is known as urethral meatus. So please make sure you're writing down the list properly. You'll get marks for this. Now next question. Mention the 2 functions of Sertoli cells. Oh, I didn't tell you about Sertoli cells, right? So basically what happens in Sertoli cells is that the seminiferous tubules which are there, my dear children, inside that you get 2 types of cells. One is the Sertoli cell that we have over here. Okay, and the other one is basically the spermatogonia. The spermatogonia, which is going to form the sperms. So Sertoli cells, what does it do? It basically nourishes the sperms. It helps in the process of spermatogenesis in a whole. Okay, it will be helping in the formation of sperms, as well as it will be providing nourishment as well. Now if you know spermatogenesis, there is a step in spermatogenesis where what happens from one spermatogonia, at the end basically you are getting 4 spermatids. But spermatids are round structure. So you have to basically transform them into elongated structure which is known as spermatozoa. Okay, which is kind of elongated. Now this process is known as spermiogenesis. What do you call it? Spermiogenesis. Spermatogenesis means the formation of sperms as a whole, all the steps which are there. Spermiogenesis means just this step, that is spermatids to spermatozoa transformation. So they were like kids, they were immature, now they are mature. You can think like that. So in spermiogenesis, my dear children, in spermiogenesis process exclusively you need more amount of hormones, yeah, and to bind these hormones together over here, Sertoli cells plays a major role. It plays a major role. So I'll just say that Sertoli cells helps in the process of spermiogenesis very much. Okay, so 2 functions: provide nutrition. It provides nourishment to the developing sperms in the seminiferous tubules. Second, it secretes factors for spermiogenesis. So they secrete certain factors necessary for the process of spermiogenesis. What does these factors do? They are known as androgen binding proteins. These factors are known as androgen binding proteins. And what are the androgens, my dear children? The male sex hormones. And what is the most famous one? Testosterone. So the testosterone, when it comes inside the testes, basically inside the seminiferous tubules, my dear children, over here. Okay, not inside the testes, inside the seminiferous tubules, when it reaches, it does not know where to go and act. So basically Sertoli cells will be releasing androgen binding protein, and testosterone will be binding over here. Coming and binding towards this particular region, my dear children, would be in more amount in this particular region, and hence in the process of spermiogenesis. Okay, it helps in the process of whole thing, that is spermatogenesis in a complete sense, but majorly spermiogenesis, because this is there in your NCERT textbook. Okay, will you remember these 2 points? You have to remember. Okay. Now next point, what is the number of chromosomes in the following cells of a human female? So we have primary oocyte, we have secondary oocyte. What are this? Basically, I tell you. So in the females we have oogonia. We have what? Oogonia, which is the one that will be further dividing and forming the primary oocyte, and this primary oocyte will divide to form secondary oocyte will divide to form, let's say, ovum at the end, the female egg, the female gamete which is there. Now we have oogonia. The oogonia will be increasing the number with the help of mitosis, my dear children. They will increase the number. It is there in the fetal ovary. So basically when I was a small fetus inside my mother's uterus, my dear children, at that particular point only, I had so many oogonia, and this oogonia increased the number way too much. They increased the number way too much, and one among them basically, few among them were growing into something that is known as primary oocyte. That is known as primary oocyte. So until now, the number of chromosomes was 46 only, just like any other cell in my body. It was 46. But now, from this point onwards, primary oocyte has started division. What division? Meiosis. Meiosis 1. I told you meiosis happens in 2 stages. In meiosis 1, this primary oocyte will now divide into 2 cells. One is secondary oocyte, and this is the polar body. So in the secondary oocyte, you have 23. So halving the number of chromosomes happens over here. After meiosis 1, always reduction happens. So they will be haploid. Secondary oocyte and the first polar body will be haploid. So this is an unequal division, which is why you get first polar body and then secondary oocyte. This cell is not going to divide further because it is of no use. It might get degenerated as well. So now you just have secondary oocyte. It will also undergo division because you have still meiosis second remaining, right? It will undergo division, that is meiosis second. And now what do you get? You get ovum, my dear children, and second polar body. And the second polar body. Again, will you reduce the number of chromosomes? No. It has become haploid over here. We just keep it haploid here also. So ovum is also haploid. The sperm is also haploid. Haploid sperm, haploid ovum comes together to form diploid zygote. Now you are asked what is the number of chromosomes in the following cells of a human female. That is primary oocyte, my dear children. Primary oocyte is just like another cell of our body. It will be having 46 number of chromosomes, that is diploid 2n. What about secondary oocyte? They have completed division, at least meiosis 1. So it will be half. So 23 chromosomes will be there over here. Always remember, my dear children, always remember that after puberty, okay, when we reach puberty, that's when the primary, so all the cells which are there, okay, all the cells, all the primary oocyte which is there, it is kind of arrested in this stage, meiosis 1. It has not completed its division. But when it reaches puberty, when we reach puberty, my dear children, that's when certain hormones will be coming into play, and finally this primary oocyte will be completing its division to what? Secondary oocyte. Now the secondary oocyte will try to again initiate the next meiosis 2 cycle, but then what happens is that it gets stuck there. It gets stuck there. Finally, it will complete its meiotic division only after fertilization. When fertilization happens, at that point the secondary oocyte will finally divide into what? Second polar body and the ovum. And the ovum and the sperms which have come to fertilize, they will be fusing together. Always remember that it's after fertilization, after. Okay, after basically when both of them comes together, that's when ovum is formed. I hope you remember that. Now, next thing, differentiate between spermiogenesis and differentiate spermiogenesis from spermiation. So for that, I have to tell you the whole process of spermatogenesis. So are you ready? If you are ready, listen to me carefully, okay? So first thing we have is spermatogonia, pardada. It's a grandfather, okay? Now this spermatogonia, which is having 46 number of chromosomes, will be dividing into so many spermatogonia, okay? There will be so much division. Okay, division. Now what happens, basically one among them will be differentiating and saying that finally I think that I have to form sperms. Okay, so what is that one thing known as? It is known as the primary spermatocyte. It is known as the primary spermatocyte. So how do you get primary spermatocyte? These spermatogonia will divide and increase in number and also differentiate as in now it feels that I want to have a particular function. Okay, so it differentiates into primary spermatocyte having 46 number of chromosomes. Now it will start undergoing meiotic division. In meiosis 1, when it divides, you get 2 cells. Will it half or not? Yes, it will half. It will be having 23 chromosome each. Again, will it divide? Yes, it will divide because meiosis happens in 2 stages. So this time meiosis 2 happens, this cell divides into 2, this cell divides into 2, my dear children. How many number of chromosomes? Same number of chromosomes, ma'am, because only after meiosis 1 you have half the number of chromosomes. After that it's all the same. Okay, now you have all these cells. Who are they and who were they? Primary spermatocyte divided, and what did we get? We got the secondary spermatocyte. Just like that. Just like oogenesis, no, in oogenesis there is only one difference, that is when primary oocyte divides, you get secondary oocyte, but you get first polar body which is of no use, it won't divide further. Which is why after division, okay, this is a very important point, after division, okay, you will get only one ovum. So there's a very important question that comes, that is basically, suppose you have 500 spermatogonia and you have 200 oogonia, how many ovum and sperm will be formed? How many ovum and sperm will be formed? Let me explain this and I'll tell you, okay? So basically, this is the secondary spermatocyte. This divides. Now, what did we get over here? They are the immature ones, that is spermatids. They are the spermatids. Now, the spermatids which are there, they are going to transform into the mature form which is known as spermatozoa, my dear children. Spermatozoa. And what do you call this process as? What do you call this process of formation of spermatozoa? This is known as spermiogenesis. Spermiogenesis. It is like, reforming again. This is the whole process is known as spermatogenesis, but here it's kind of very special. The kids are maturing, so it is spermiogenesis. Again, we are keeping it a name and giving spermiogenesis. After that, after they matured, finally they would be released out from the seminiferous tubules. This was all happening in the seminiferous tubules. Finally, they will be releasing out to the seminiferous tubules, and this process is known as spermiation. It's like aviation, okay? So they would be finally moving out, you can remember like that. Out of the seminiferous tubules, now they are going into accessory ducts, that is rete testes and everything like that. Okay, I hope now you understood the difference, okay? Now, spermiogenesis, it is the process where spermatids transform into spermatozoa. It involves structural changes like the formation of acrosome, flagellum and nuclear condensation. So basically all the things that is to make the sperm mature, okay. Acrosome as in at the head region of the sperm, my dear children, there's a enzyme filled thing, so that is being formed. Apart from that, a long flagella is being formed, the tail, okay, and then the nucleus which is there, it also condenses to one particular location just below the acrosome, the head region. You get it? So that all things happens over here. What about spermiation? It is a release of mature spermatozoa from the seminiferous tubules into the reproductive tract. Into the reproductive tract. So basically, you can don't write this, write accessory glands. Accessory ducts. Right, accessory ducts, that is rete testes and everything. It marks the final step of spermatogenesis before sperm enter the epididymis for maturation and storage. Don't you think it is the final step of spermatogenesis? Yes. Before the sperm enter the epididymis for maturation and the storage. So just write this, you get marks. Okay, now the question, let me return to the question. So 500 spermatogonia and 200 oogonia is there. How many ovum and how many sperms are being formed? Listen, all the spermatogonia which will be basically transforming into the primary spermatocyte, all of them, one primary spermatocyte can give rise to 4 sperms. So if 500 of them are there, so 500 into 4, my dear children, it will be 2000 sperms will be formed. But what about the 200 ovum? Oogonia. 200 oogonia, supposing that all of them are forming the primary oocyte. If 200 of them are there, then at the end, from one primary oocyte, how many ovum did you get? 1 ovum. So 200 of them, all of them, 200 will be giving 200 ovum. Not into 4, nothing into 4, nothing into 3. So it will be just 200 ovum. I hope you understand the difference, because it is getting lost 2 places, you know. So which is why this is not happening. And this is a very important question. Now, mention the roles of LH and FSH during spermatogenesis. LH and FSH. What do you mean by LH? LH means luteinizing hormone. What do you mean by FSH? FSH stands for follicle stimulating hormone. And what are the roles of both of them during spermatogenesis? That is a question. I'll tell you. So listen to me carefully. So over here, this is a seminiferous tubule. Imagine. Are you imagining? Yeah. This is also seminiferous tubules. In between 2 seminiferous tubules, you have a space which is known as interstitial space. Interstitial space. Okay? Agreed? Now, this interstitial space contains certain cells which are known as Leydig cells. Leydig cells. Cool? Now, inside the seminiferous tubules, what we have? We have Sertoli cells and we have spermatogonia. Agreed? What is going to happen is that, you know, we have hypothalamus in our brain. There is a region that is known as hypothalamus. And when men attains puberty at this point of time, this hypothalamus is going to secrete something that is known as gonadotrophin releasing hormones. What? Gonadotrophin releasing hormones. That means there are 2 hormones which are known as gonadotrophins. So we're talking about that. So that we have to release both the gonadotrophins. That is the thing. But who are they going to say all this thing to? Gonadotrophin releasing hormone is going to act the pituitary gland, my dear children. It is going to act on the pituitary gland. This is what, and pituitary has 2 lobes. One is anterior lobe, one is posterior lobe. This is the anterior pituitary on which the gonadotrophin releasing hormone will come and act. What happens now? The pituitary gland says that, I'm going to release 2 things. Since you are saying so, one is LH and the other one is FSH. Okay? LH, luteinizing hormone, and FSH. Fine. Then what happens? This luteinizing hormone is going to act on the Leydig cells. It acts on the Leydig cells and makes the Leydig cell release certain androgens, that is testosterone, my dear children. Okay, now androgens is being released. What about FSH? I'll tell you about FSH. So androgens are being released. Spermatogonia are basically trying to divide and help in the formation of what? Sperms, my dear children. But this process requires androgens. So these androgens which are there, which are being formed, they go over here inside the seminiferous tubules and helps in the formation of sperms. But entirely they cannot do it alone. They need the help of someone else also. They are not like, they want to like roam around. They don't want to stay at one place. So there should be someone who could hold them at one place. Who is that? It is androgen binding factors or androgen binding proteins. Okay, and who releases it? It is the Sertoli cells. Who tells Sertoli cells to release all this thing? So basically it is FSH. FSH which is there, it goes and acts on the Sertoli cells, and the Sertoli cells will now release certain factors. In your NCERT textbook it is just written factors, so you can remember at factors. Now these factors would make the androgen stay at one particular place and help in the whole process. Basically, specifically if you are saying, it majorly helps in a process that is known as spermiogenesis. I told you, where the spermatids are converted into what? Spermatozoa. I hope you got it. First, the hypothalamus is there. It releases gonadotropin releasing hormone. This gonadotropin releasing hormone, that means you're going to tell the gonadotropins to release, you're telling the gonadotropin hormones to get released. That is what you mean. Okay, and who releases the gonadotropin hormones? It is the pituitary gland. Pituitary gland has two gonadotropins, that is FSH and LH. Okay, first thing, LH goes and acts on the Leydig cells and releases androgens, and these androgens, when they go up there, they do not know what to do, where to go and everything. So there is someone who guides them. What is that? ABP, androgen binding proteins, or basically factors, which are released by whom? Sertoli cells. Who says the Sertoli cells release all this thing? FSH. So you get it? If this guy is not there, ABP is not there. If ABP is not there, this guy cannot come over there and just stay there. You get it? And hence the whole process would not be happening. So this is the role of LH and FSH. Luteinizing hormone acts on the Leydig cells, interstitial cells, and stimulates them to secrete testosterone, that is androgens. Okay. Now testosterone is essential for spermatogenesis. The whole process of spermatogenesis is very much essential. If we talk about follicle stimulating hormone, it acts on the Sertoli cells and stimulates them to secrete some factors that aids in what? Sperm cell maturation, that will help in the sperm maturation, helps in the process of spermiogenesis, conversion of spermatids into spermatozoa. Will you be able to write this? It's easy, no? If you remember it like a story, you will be able to remember it. Okay? So this is the Pardada, okay, this is the grandfather, this is the grandfather, and these are the chutku children that they have. Okay? Now, how is oogenesis markedly different from spermatogenesis with respect to the growth till puberty in the humans? Okay, when does it start? Oogenesis, I told you, it starts before birth, before I am born, my dear children, when I am in the mother's womb. At this point of time itself, in my ovary, the oogenesis have started. The formation of oogonia, basically, the primary oocyte have been started. It is stuck there, it has not underwent the meiotic division completely, but it's just stuck there. But this formation is definitely happening. What about spermatogenesis? At puberty. At puberty only spermatogenesis will be starting, only when the Pardada hormone says that you have to release something. Okay. Now pause before puberty. So it stops at an early stage and remains paused until puberty. I told you, it stops, right? When we are baby, at that point itself it's stuck and remains paused until what? Puberty is being reached. Does not start at all before the puberty. Right? It does not start at all before the puberty. Now, number of cells produced. Only one ovum is produced from one primary oocyte, whereas 4 sperm cells are produced from one primary spermatocyte. You know that, right? Now, process happens once a month after puberty. In the case of human beings, okay, every month, happens every month, right? Oogenesis will be happening once a month. Did I say every month? So once a month after puberty it will be happening. It happens continuously after puberty, any time, any moment it can happen. Now stops or continues. So stops at menopause. So what happens, basically the formation or oogenesis will be stopping at the last thing, that is menopause. Menopause means the cessation of the menstrual cycle. It stops around that. So it's almost around 45 to 50 years where it will be stopped, whereas it continues throughout life. That is why we say the females won't be fertile after some point. When they are stopped releasing the eggs or making the eggs, how can they be fertile? They cannot give rise to the young ones. Whereas males, they are always evergreen, so they can be producing children at whatever age is there. So these are the differences that you should definitely know. Please not miss certain differences. If you have any doubts, children, always I say, even if I am teaching like very in a broad spectrum, I still feel that at least some of you might be having doubts, and very okay to have doubts. You can ask me in the comment section. Please feel free for that. I am here to help you out in your examination. Okay, so please feel very free for that. Okay, so when I was doing offline classes at that point of time, children used to call me at even 12 o'clock, 1 o'clock for asking doubts, and I used to explain as well. So that's the relation I have with children. Same thing I have with you guys, even though we do not see but yes, I definitely know that I should be helping you guys. So if you have any doubts, please put it down. If it's in my range, I'll definitely help you out. So mention the fate of corpus luteum and its effects on the uterus in the absence of fertilization of ovum in a human female. I'll be talking about this a bit later. So question number 7, we can keep it for some time, because I want to talk about menstrual cycle first before going ahead with that. So list any 4 hormones secreted by placenta. Placenta. What are the 4 hormones secreted by placenta? It is human chorionic gonadotropin, human placental lactogen, progesterone and estrogen. Don't think that progesterone and estrogen are released by ovary, ma'am. Yes, it's okay. It's okay. It is also being released by placenta when once placenta is being formed. So there are 4 hormones. Never forget it. Nothing else. Now, human chorionic gonadotropin, it is the one that maintains the corpus luteum and supports the pregnancy. Why do I say so? Why do you need corpus luteum? What is corpus luteum? So, corpus luteum, as of now, if you do not know anything about corpus luteum, just remember, corpus luteum is something that will be releasing hormones, right, progesterone and things like that, which will be maintaining the endometrium. So if corpus luteum is not there, there is no progesterone. There is no progesterone, it is not going to maintain the endometrium. If the endometrium is not maintained, the embryo which is there, it cannot come and implant. It won't get nourishment. So to maintain the corpus luteum, you need something, and that is human chorionic gonadotropin. So during pregnancy, when pregnancy happens, at this point of time, before there was someone else, it was luteinizing hormone which was responsible for maintaining the corpus luteum. Okay, it was responsible for maintaining corpus luteum. And as I said, it releases progesterone. But this progesterone do not want LH to control all this thing. So it says that, who is the pardada of LH? GnRH. It goes and says the hypothalamus to stop the secretion of GnRH. And if GnRH is stopped, LH is also stopped. If LH is stopped, corpus luteum will be not there. If corpus luteum is not there, this guy also won't be there. So this guy does not know about that. So there should be someone. Usually what happens, this guy goes, says that, and all this happens, and there is no progesterone at the end, and the endometrium sheds off. Endometrium, it sheds off. That's why we get menstruation. But at this point of time, since pregnancy is happening, we do not want it to shed off. We want someone else to control over it. Who is that corpus? Who is that? Human chorionic gonadotropin, my dear children. It maintains the corpus luteum and supports the pregnancy. Now you have human placental lactogen. It helps in the fetal growth, that is the growth of fetus, and maternal metabolism regulation as well. Maternal metabolism regulation as in the carbohydrate breakdown, making up of things, proteins, all the things, breakdown, making all addition, subtraction, everything is being handled by placental lactogen, human placental lactogen. Okay. Now progesterone is there. It maintains the uterine lining and prevents contraction during pregnancy. Don't need to remember all those big things. Just remember it maintains the uterine lining. Even estrogen, it helps in the maintenance of uterine lining. It also supports fetal development. That's all. It's okay. You have to remember that. Now, let me tell you one more question. What are the other hormones which increases in the blood of the pregnant female? Only during pregnancy, these hormones level increases way too much. What are those? Please remember, it is cortisol. Okay, cortisol, and then you have prolactin. You have prolactin and thyroxine. So thyroxine and all is usually being produced, even cortisol is usually, but it is, okay, so if at all the question comes like this, I will reframe the question. What are those hormones whose secretion increases many folds in the maternal blood during pregnancy? So it will be cortisol, prolactin and thyroxine. Thyroxine helps in metabolism, prolactin helps in the mammary gland formation and things like that, and cortisol has its own sexual functions as well. Okay, and there is also one more hormone that is released during pregnancy, during the later phase of pregnancy by the ovary, and that is relaxin. So I hope you remember all these things. Only 4 hormones for the placenta, that is this. Okay, one hormone towards the later phase of pregnancy, during the end of pregnancy, it is relaxin produced by the ovary. Please remember, it's not placenta. Okay, children get confused. Now, what are the hormones that gets increased many folds? It is cortisol, prolactin and thyroxine. Works? Chalo, next thing. Unless fetal ejection reflex is produced, normal parturition does not occur. Substantiate the statement. What do you mean by this question? Basically, they're saying that, you know, fetal ejection reflex. What do you mean by fetal ejection reflex? Basically, the baby which is there, for example, I am the baby and I am inside the uterus of the mother. Okay, so when I fully develop, okay, what do I do? What do I do? Basically, there will be certain hormones being released by me, and then, or basically, I can just say that there are certain changes happening in the uterus. What do? I try to sneak out. And when I try to sneak out, if you know this is the uterus, and you have a cervix region, my dear children, and on the cervix region, there are so many receptors. There are so many receptors which are there. Okay, so when basically I try to sneak out, these receptors will become highly active. It becomes what? Highly active, my dear children. The neurons, whatever information is there, this information, the stress receptors, when they are active, they give information to the pituitary gland of the mother. They give the information to the pituitary gland of the mother. And what does the pituitary gland of the mother do? It will be releasing something that is known as oxytocin. It will be releasing something that is known as oxytocin. Now, this oxytocin is very much important. Why? Because what happens, only if this oxytocin is there, that's when it will allow contraction and relaxation of the uterus. But how does it happen, ma'am? So basically what happens is that it's not just the baby which is trying to move out. Okay, there are so many changes that is happening inside also. So inside the uterus, in the placenta also plays a major role. In the placenta, the amount of estrogen will be released way too much. Okay, estrogen which is there, it is also released way too much. And this estrogen, when it is released, what happens? You know, uterus is made up of 3 layers, and the middle layer is known as myometrium. The estrogen will be acting on the myometrium and making it to show all the oxytocin receptors, to basically show up all the oxytocin receptors. And now when the baby moves, tries to move out, the stress receptors gets active. This gives a neural signal to the pituitary gland. From the pituitary gland, a hormone is released that is known as oxytocin. This oxytocin will come and bind over here. Once it comes and binds over here, what will happen? It is a myometrium. Myometrium is made up of smooth muscles. It will start to contract, relax, and the baby will be finally moving down. So unless fetal ejection reflex is produced, do you think the stress receptors can get active? Do you think it can get active? Or even if it gets active, the placenta is not releasing. The placenta, which is also a part of fetal ejection reflex, it is not releasing the estrogen and making up so many receptors active over there. Do you think the oxytocin can come and land over here and do the contraction? No. So that is why fetal ejection reflex is very much important. It is a neuroendocrine mechanism that triggers what? Labor. So fetal ejection reflex is from the fully grown fetus as well as the placenta. Please write like that. And is a neuroendocrine mechanism that triggers the labor. It is initiated by mild uterine contraction due to full-term fetal growth and pressure on the cervix and further activation of what? Receptors. So now the receptors are activated. This is also moving down. So by the pressure on the cervix and the further activation of receptors, everything happens. Why? Because of full-term fetal growth, my dear children. This stimulates the release of oxytocin from the maternal pituitary gland. Oxytocin induces stronger uterine contraction, which further enhances the cervical dilation. The cervix will be dilated. A positive feedback loop is created where more oxytocin. What will happen? This will release more oxytocin, more contraction, more oxytocin, more contraction. So this happens until and unless the parturition happens. Without this reflex, normal delivery cannot occur naturally, my dear children. That's why we wait for there should be some signal. That's when the normal delivery starts up. You get it? Otherwise, you induce it. So this is the whole thing. Please remember, it's a neuroendocrine mechanism. Very, very important question. Okay. So you can write off starting with this particular question if you feel it's too confusing for you to write placenta, this and that. So it is initiated by mild uterine contraction due to full-term fetal growth, okay, which puts pressure on the cervix, and also the placenta which releases estrogen and helps in activation of receptors. So two things comes together to give the fetal ejection reflex. Further it is transferred. Always remember, pituitary gland, as I said, there are 2 lobes, anterior and posterior. So this is the posterior lobe that gets active. This is what? The posterior lobe that gets active, my dear children. Cool? Now going ahead and releases oxytocin. So this is the thing. Now trace the path of spermatozoa from the testis up to ejaculatory duct. I told you already, we studied this, but let's see. So this is, suppose the testis, you have the testicular lobules. Just check through the answer, okay? The testicular lobules has seminiferous tubules. The seminiferous tubules will be opening up and forming the rete testis. the rated testis will be opening up to give vasa efferentia. It is finally exiting, E for efferentia, E for exiting, into the middle of that. And but then later it understands that, oops, I am getting stuck over here. So it forms something like this, becomes stuck over here for some time. This is known as epididymis. What? Epididymis. From epididymis, the sperms will be going up with the help of vas deferens. With the help of what? Vas deferens. And now, suppose this is a urinary bladder that we have over here. The vas deferens which is there, it will be receiving a secretion from another gland which is known as seminal vesicle. Both of them will now be opening together, and this is known as ejaculatory duct. This is known as ejaculatory duct. This ejaculatory duct will be opening into the urethra, which ends up until the end of the penis which is there over here. You get it? So, seminiferous tubules, sperm's production occurs in the testis. Rated testis, the sperm moves from the seminiferous tubules to the rated testis. Sperm passes through the vasa efferentia into the epididymis. Epididymis, sperm matures and it's stored here. Yes, epididymis is a place, it is 6 meters long if you put it like this. So, it is a place where actually the sperms mature and is stored over here. Okay. Now, sperm travels from epididymis through the vas deferens. Don't think that sperm maturation happens in the seminiferous tubules, ma'am. You told us about spermiogenesis. But there are a lot of other, processes still yet to be happened. So it happens in the epididymis, and then vas deferens takes it and then goes with the ejaculatory duct. Sperm mixes with secretions from the seminal vesicle and enters the urethra. That's all. Easy. Okay. What's the time now? Okay. When and where do chorionic villi appear in human state their function? What do you mean by chorionic villi? So, my dear children, when zygote is formed, you know how zygote is formed? Male gamete, female gamete, zygote is formed. You get one cell. This one cell will further divide into two. This one cell which is there, it will further divide into two. This two cell will divide into four, and things goes on. So, these are embryonic stages that happens. Okay, these are embryonic stages that happens. And now you reach one stage which is known as blastocyst stage. What is so special about blastocyst stage? So, blastocyst stage is something like this, where the outer cell that is there, it is known as trophoblast. It forms something that is known as trophoblast. The inner cell which are there, they are going to form something that is known as inner cell mass. Inner cell mass. Okay. The trophoblast which is there, and I know it is and this is the stage where it is going to get implanted in the uterus. This is the stage where it is going to get implanted into the uterus, my dear children. So, suppose this is a uterus, the blastocyst comes over here. This is the endometrium that we are having. The blastocyst comes over here, my dear children, slowly gets embedded there, slowly gets embedded there. The outer layer is trophoblast, inner is inner cell mass. The outer thing which is there, it will slowly form finger-like projections. It will slowly form finger-like projections, and from the maternal side also some tissues will be coming and joining with all these things. So, they are going to form a structure together. That is the trophoblast, the trophoblast which is there, it's formed finger-like structure, and also the other maternal tissues are also going to form something over here, and together they are going to form something that is known as placenta. What do they form? They form something that is known as placenta. And what happens to the inner cell mass? Now, the inner cell mass is the only thing that is left, that becomes the fetus, that grows into the fetus. Okay, that will be something that will be growing into the fetus. So, this finger-like projection that I was talking about is the chorionic villi. So, chorionic villi appears during the early stages of embryonic development around the 2nd week of pregnancy. Around what? The 2nd week of pregnancy. You don't have to write that, okay, because it is not there in your book. Now, they develop from the trophoblast, outer layer of the blastocyst, and extend into uterine tissue. The maternal tissue is basically the uterine tissue. Now, function. It helps in exchange of nutrients, gases and waste between the mother and the fetus. Yes. And it facilitates implantation by anchoring the embryo to the uterine wall. Yes, obviously it anchors first, then it helps in exchange, and plays a major role in the formation of placenta, ensuring what? Fetal development. So, how you can frame this? First thing, you can write this. It plays an important role in what? In the formation of placenta, ensuring fetal development. Then you say it helps in facilitating implantation by anchoring the embryo to the uterine. Okay, basically what you can do is, instead of putting this as first point, you can put this as first point. This is the first point. It facilitates what? Implantation. Chorionic villi will be facilitating implantation. It can get into and settle down in the endometrium layer. So, it facilitates implantation by anchoring the embryo to the uterine wall. It anchored there. Now, it will slowly help in the formation of what? Placenta. Then, it helps in the exchange. If placenta is formed, it will obviously help in the exchange of what? Nutrients, gases and waste between the mother and the fetus. So, these are the functions that you can possibly write. Will you be able to write? Will you be able to write it? Yes, you will be. I trust you completely, 101%. Okay. So now study the following chart. Name the hormones involved at each step. Explain their functions as well. So hypothalamus, pituitary, testes and sperms. I already told you, hypothalamus releases gonadotropin releasing hormone, pituitary releases FSH and LH, testis to sperms. So testis to sperms, who is there over here? Testosterone. It is testosterone. It acts on the from the pituitary, FSH and LH comes, and then it acts on the testis. The testis will be releasing testosterone. Yes, it is testosterone. Yes, fine. Okay. So how do you write it? GnRH produced by the hypothalamus. It stimulates the pituitary to release what? LH and FSH. LH produced by the pituitary gland stimulates the Leydig cells to produce what? Testosterone. FSH, which is there, it is produced by pituitary gland, stimulates the Sertoli cells to support spermatogenesis. Testosterone, which is there, it is produced by the Leydig cells. Yes, Leydig cells which are there in the testis, and it is going to help in the formation of what? Sperms. And also in what? Male secondary sexual characteristics as well. Easy question. Are we increasing our confidence or not? Are we losing our confidence or increasing our confidence? Let me tell you one thing. If you are not able to solve this question by yourself, when I tell the question, do not feel that you should be sad about it. Why? Because every time you basically do not know anything, after 1 minute or 2 minutes, you are basically understanding the thing now when I am teaching, right? So this is your victory. Do not think that you're losing out on anything. Children, you are going to ace the exam. Trust me on that. I am telling you again and again. Now, trace the development of oocyte till ovulation in human female reproductive system in the menstrual cycle. So, oogenesis begins in the fetal stage. Agree? Oogonia is formed in the ovaries. None of them are added after birth, my dear children. Now this oogonia which is there will transform into primary oocyte, and it is arrested. This primary oocyte which is there, it is arrested in the prophase 1 of meiosis, surrounded by granular cells and forming the primary follicles. I'll tell you once more what happens in the oogonia. The oogonia which are there, they will differentiate slowly and they will form primary oocyte. The primary oocyte, it will start off, it will try to do meiosis, but it gets stuck there. In what phase? Prophase 1 stage. So meiosis, it has prophase, metaphase, anaphase, telophase. So in meiosis 1, you call it prophase 1, metaphase 1, anaphase 1, telophase 1. This is what you call it. Right. So I am saying it entered primary oocyte, it will enter meiosis, then that's how it forms the secondary oocyte, right? But it gets stuck there at prophase 1 stage in the embryo itself, my dear children. But at this point of time, the primary oocyte is covered by certain cells. The primary oocyte which is there, it is covered by certain cells. You call them granulosa cells. What do you call them? Granulosa cells, and together the whole structure you call it as primary follicle. Cool. Now what happens? Follicle reduction. So many degenerate, many follicles which are there, my dear children, it will be so many follicles will be there, and so many of them will be degenerating. But only 60,000 to 80,000 will be remaining at puberty. Only 60 to 80,000 remains at puberty in each ovary, not in one ovary. In each of the ovaries, 60 to 80 will be remaining. Now what happens? The secondary follicle forms. More granulosa cells are being added up over here. More granulosa cells are being added up over here when you hit puberty, at that particular point of time. So more granulosa cells are added, and one theca is also developed. One extra layer is also developed, which is known as theca layer. Now you call this whole structure as what? Secondary follicle. You call this as secondary follicle. But inside secondary follicle also, what do you have? You have the primary oocyte only, which is still stuck at prophase 1 stage. Now tertiary follicle forms. What is the difference between tertiary follicle and secondary follicle? That is, basically, there will be a fluid-filled cavity forming over here, my dear children. There will be a fluid-filled cavity forming over here, which is known as antrum. And what about the theca? The theca will be dividing into inner theca interna and outer theca externa. Theca layers which are there, they are dividing like that. Okay. Now you have this, and let me tell you, this is when tertiary follicles forms, and this is the point where finally it will undergo all the division and form what? Secondary oocyte. So inside the tertiary follicle, what do you find? You find the secondary oocyte finally. So meiosis 1 completes, forms secondary oocyte. That is, there is one haploid secondary oocyte being formed, and there is one first polar body also. The first polar body which is there, it gets degenerated. It won't be there. It will get what? Degenerated, my dear children. Now going ahead, what happens? So we have the secondary oocyte, the polar body gets degenerated, and we have granular cell layers, we have antrum, we have theca interna, theca externa, everything. Now slowly there will be formation of Graafian follicle. So the tertiary follicle matures into what? Graafian follicle. Okay, and in Graafian follicle one very important thing happens, that is basically it enlarges in size obviously, and the secondary oocyte for the first time, remember it's a very important question, MCQ question, a very important thing forms over here from the secondary oocyte, and that is known as zona pellucida. So very important layer is being formed, and this layer which is being formed, my dear children, it is known as zona pellucida. Okay. Outside zona pellucida there is one more layer, but it's not formed by the secondary oocyte. It is known as corona radiata. It is known as corona radiata. So your secondary oocyte looks like this: secondary oocyte plus zona pellucida plus corona radiata, and then all the other cells which are there. But the secondary oocyte releases this in the Graafian follicle. Okay, now Graafian follicle is the mature one. So whenever we say that each time ovulation happens, one follicle, one primary follicle will be basically converting into going through all the stages, secondary, tertiary, and then Graafian. Only one will be forming in the Graafian follicle. The Graafian follicle will rupture now, will be rupturing and releasing what? The secondary oocyte. So in which stage is the thing released? In the case of ovulation, in the secondary oocyte stage, my dear children. Still it has not divided completely into ovum. This also is stuck. The secondary oocyte is also stuck in meiosis phase 2. Okay, in meiosis 2 it is still stuck there. When will it complete the division? When the sperm will come and fertilize. When it tries to fertilize, at this point of time, finally it will complete its division and form the ovum. So ovum and the sperm will finally come together, the nucleus of it. So ovulation occurs, Graafian follicle ruptures, releasing the secondary oocyte, meiosis 2 completes, only if fertilization occurs. Okay, so this is the developmental oocyte till ovulation. I hope you got it. Next, so this was a 5 marker, right? Yeah. Now, describe the various events that occur during menstrual cycle. Yes, after this we will discuss the 7th question and then we will go ahead. So, describe the various events that occur during menstrual cycle, my dear children. Here you have to have this remembrance of the picture. Listen to me carefully, okay? The egg and the sperm, when they meet, you get zygote. The zygote divides itself and form embryo. Now, this embryo will need someone to provide nutrition to become a fetus, right? Who is that? Who is that someone who provides nutrition? It is the endometrium layer of the uterus, the innermost layer of the uterus, which is endometrium. That is the one that provides nutrition. But what happens is that, suppose the fertilization has not happened, suppose zygote is not formed, embryo is not formed, the weight is waste. The weight of the endometrium, it is a waste. It will slowly shed down, and this is known as menstruation. What do you call this? Menstruation. So I will say, suppose this is day 1. On day 1, menstruation happened. That means the blood has been lost. The endometrium layer which was there, it is now shedding down. Okay, it is now shedding down. That happens over here for almost around 1 to 5 days. For almost around 1 to 5 days, this thing is happening. Okay, so now endometrium is very thin because we do not have it, all of it shedded down. Now parallelly, what will happen? The body thinks we should be trying it once more. Let's see one more time. Every month it thinks like that, and every month what happens is that the primary follicles, you remember, which was there in the ovary, they are now gonna mature into secondary follicles, and secondary follicles are gonna mature into Graafian follicle. But not all of them will mature together. Initially they start off by a lot of them maturing, but after a point someone realizes that, hey, I am the dominant one, I should be the one that is maturing. Please do not mature. Please guys, you do not mature. So basically the follicles would be maturing. One among them would be maturing into what? The Graafian follicle. The Graafian follicle. Remember, there is first phase which is known as menstrual phase, where the blood is getting lost. The endometrium is getting shredded down. Now, once it ends, almost around 1 to 5 days after it ends, what happens? Slowly, the follicles which are there, it starts maturing. Now we say that we have entered the next phase, which is follicular phase. Parallelly, one more thing is happening. I told you there are granular cells in the follicles. These granular cells are the ones that will be releasing some estrogen. Some what? Estrogen molecules, some estrogen hormones. Now this estrogen, what will it do? It will slowly reform the endometrium. So the endometrium which is shut down, it is starting to reform. The follicles basically are maturing parallelly. Okay, so this is known as follicular phase, and it is also known as proliferative phase because the endometrium is what? Proliferating. It is being made up. Now at one point of time, please remember one very important thing. Over here, when I say follicles are developing, who are developing these follicles? What are the hormones behind it? It is FSH, follicle stimulating hormone, and a little bit of effort can be given to LH, luteinizing hormone as well. And who are the ones who release them? It is the pituitary gland that release them, my dear children. Pituitary gland is releasing FSH and LH. Please remember it. Okay, this question can come. A and B hormones are released from what? Pituitary gland. Now over here, estrogen which is there, it is released from the follicles. Right? And where are the follicles? In the ovary. So can I say estrogen is an ovarian hormone? Yes. So estrogen is an ovarian hormone. So these two things are happening. Now at one point, my dear children, what happens? The amount of estrogen being released by these granulosa cells are way too much. So there is a spike here. There is a spike here. And when estrogen does have a spike, at the same time LH also getting influenced by this, it also has a very big spike. The luteinizing hormone increases way too much. Okay, on the 14th day of the cycle. Assuming that we are starting the cycle from first, on the 14th day what is happening? There's a spike happening in LH. You call this as LH surge. Now when LH surge happens, the graphene follicle which is there, it will burst open to release what? What is there inside the graphene follicle? Ovum? No, it's not the ovum. It is the secondary oocyte which is stuck in meiosis 2. So secondary oocyte will be released. Yes, and this process is known as what? Ovulation, my dear children. So there's a phase here which is not mentioned over here. It's known as ovulatory phase. Okay, now what happens? Whatever thing is left behind in the graphene follicle, whatever things are left behind, what do you call it? You will call this as corpus luteum. You will call this as corpus luteum. And what does corpus luteum do? Corpus luteum releases two hormones. Again, it releases estrogen and majorly progesterone. So until now, progesterone was like this. But when corpus luteum came into action, the progesterone level increased. Well, there is a reason for this. What is the reason? This progesterone is the one that will increase what the endometrium. It is the one that maintains the endometrium. It is the one that will be making the endometrium more thickened, my dear children. And why are we doing all this thing? Because the egg has released. Sperm can come anytime. Fertilization can happen. There should be someone to basically what? Nourish it. That's why the endometrium has been made. And who makes it? Progesterone and estrogen. Okay, and both of them, they would be released from the corpus luteum itself. Okay. Under whose command? It is LH. It is luteinizing hormone which basically did all this thing, and luteinizing hormone is the one that will be maintaining the corpus luteum, and corpus luteum will be releasing progesterone and estrogen. Okay, and they would be now, since the progesterone level increases, what will happen? Endometrium increases way too much, and even estrogen will also increase way too much. You see a dip there, but then it slowly increased, right? Now the problem comes that basically slowly what will happen? The progesterone which is there, it will go tell the GnRH, gonadotropin releasing hormone, to stop the secretion of gonadotropin. It does not know that gonadotropin is going to say LH that you should also stop. If this guy stops its secretion, obviously the pituitary hormones will also stop its secretion, so LH is not released. If LH is not released, corpus luteum would not be there. If corpus luteum is not there, my dear children, will progesterone be there? No. Progesterone is not there. So what will happen? And this happens really slowly. It does not happen really fast. It happens slowly. The corpus luteum, everyone waits for some time thinking that fertilization will happen now. But if it does not happen, this is what happens. The progesterone will be telling GnRH tells LH, if LH is not there, corpus luteum is not made. If corpus luteum is not there, progesterone is not there. If progesterone is not there, this endometrium which is there, it will slowly shut down, because this had the major function of maintaining it. It will slowly shut down. And again, we are starting with the next menstrual cycle. Next phase. So over here I didn't mention what do you call this phase where corpus luteum and everything is formed. It is known as luteal phase. It is known as luteal phase, and it is also known as secretory phase. Why secretory phase? Because at this point of time endometrium is going to secrete a lot of things that is going to nourish the embryo if at all it comes on. Okay. Now again you have the same cycle repeating again and again. I hope you got it. What do you write for this question now? Menstrual phase is the shedding of the endometrial lining due to lack of fertilization, drop in progesterone and estrogen levels that you see, obviously. Okay. Now follicular phase is proliferative phase as well. Now FSH stimulates the follicle growth. Follicle secretes what? Estrogen. Endometrium regenerates and thickens because of what? This estrogen. LH surge triggers what? Ovulation. At the end, when the estrogen level goes up, what happens? Slowly LH also goes up. Okay, there's a positive effect by the estrogen to the LH. So LH also goes up. There's an ovulation that happens. So graphene follicle ruptures, releasing the secondary oocyte. High LH and FSH levels are being seen over there. Now you have luteal phase or secretory phase. Corpus luteum forms, secreting progesterone. Endometrium thickens further, preparing for implantation. If fertilization does not occur, corpus luteum degenerates. Progesterone drops and the cycle restarts again. Will you be able to write this? I hope you will be able to write this. I have almost spent 8 minutes of my time for explaining you this. I hope you understood menstrual cycle, because a lot of the children have doubt in menstrual cycle. Now, I told you I will be making you solve one question after we study the menstrual cycle. Listen, mention the fate of corpus luteum and its effect on the uterus in the absence of fertilization of ovum in human female. If fertilization is not happening, my dear children, what happens to the corpus luteum? The progesterone which is there, it goes and tells the GnRH to stop its secretion. GnRH tells LH to stop its secretion. If LH stops its secretion, corpus luteum is not maintained. It will slowly start degenerating. And corpus luteum, if it is degenerating, the amount of progesterone also goes down, and hence there will be shedding. So, degeneration of corpus luteum, if fertilization does not occur, the corpus luteum gradually degenerates. It transforms into a small non-functional structure which is known as corpus albicans. It is not there in your NCERT. It's okay. Now, decrease in progesterone level. The corpus luteum stops secreting progesterone, a hormone that maintains the uterine lining, and because of this, due to drop in progesterone, the thickened endometrial lining of the uterus breaks down. The shed uterine lining, blood and mucus are expelled from the body, marking the start of menstrual cycle, that is periods, my dear children. I hope you got it. So, one by one you say, if this is not there, this won't happen. If this is not there, this won't happen, and that goes on. I hope you got it. Okay. Now, I saw a question in this regard. What is the difference between estrous cycle and menstrual cycle? So, estrous cycle happens in non-primates, and menstrual cycle, it happens in primates. Primates as in the one that has large renal capacity and things like that, okay, like us. And non-primates means you're talking about probably dog, cow and all of them. So, I have told about a detailed difference between them in the 2 marker, 3 marker or 5 marker question video. Please go watch it. Okay, so I have given you a timetable to follow. Please follow it. It is going to be really, really helpful for you. I used a lot of brain in making that. Okay. Now, let's go on to the next question, my dear children. Sorry for making you wait for long. Okay. Now, briefly explain the embryonic development in humans. Embryonic development in humans. Okay. So, now in your NCERT, it is written fetal development because it is talking about fetus. Okay. Fetus means after some point of months, you say now the embryo is finally the fetus. But anyway, you can write this answer as well. So, first month, what happens, my dear children? The embryo will slowly have a heart. Heart is there. Okay. So, you would be hearing heartbeat after 1 month. Second month, there will be limbs and there will be digits. There will be limbs and digits. After 12 weeks means you're talking about 3 months. First trimester is done. At this point of time, major organ systems form, okay. The limbs and the external genital organs are well developed at this point of time. Now, 4th month, they have not mentioned in your NCERT. We are not going to learn it, okay. 5th month, the fetus says, until now, God, you did whatever you wanted. I want to have my own things. What are your own things, child? So, it says that I want hair on my head and also I want to move around a bit. So, basically, the first fetal movements and hair appears on the head on 5th month, by the end of 5th month. Now, 24 weeks as in you're talking about the 6th month. Okay, 24 weeks as in 6 months. So, body is covered by fine hair, eyelids are separate, eyelashes are formed. So, after this, God said that let me do what I want to do. Okay, so I'll give you hair on, you wanted hair, no? I'll be giving you hair on your body also. Okay, now your eyelids which are there, let's open it. So, eyelids are separated, the eyelashes are going to get formed as well. Now, 9 months, that is full term, the fetus is fully developed and ready for what? Birth. Finally, it is going to come out. So, first month, heart. Second month, limbs and digits. By the end of 3rd month, you have most organ system being formed over there. Limbs and external genital organs are very well developed. Now, 5th month, hair and movement. Okay, it will move around. And now 6th month, or basically 2nd trimester end, body would be covered with fine hair. Eyelids would be separate. Eyelashes will be formed. Everything is there. 9th month, baby is out. Okay. Now, if you want, you can, let's say, if you feel that, oh ma'am, I want to add up a bit in this answer, in the embryonic development, you can tell the zygote is formed first, and then what happens, the zygote undergoes mitotic division to form morula, solid balls of cells. So, morula is what? A stage of embryonic development itself. So that's why I had told you can write it. Now slowly the morula, which is 8 to 16 cell stage, it will divide further to give blastocyst. Morula develops into blastocyst, which has outer trophoblast and inner cell mass. We discussed about that, right? Trophoblast will be helping in implantation, right? Now blastocyst embeds in the endometrial lining. Slowly what will happen, the inner cell mass which is there, it will give rise to three things: ectoderm, mesoderm and endoderm. Ectoderm will give rise to skin and the nervous system. Mesoderm, the middle layer which is there, it gives rise to muscles, bones and circulatory system. Endoderm, the innermost layer, it gives rise to digestive and respiratory tracts as well. Slowly organs will be formed and the fetal development proceeds. So if you want, you can support your answer with this. Majorly, please make sure you are writing this. So instead, in one liner thing, you can write that basically zygote is formed, then zygote undergoes mitotic division to form morula, and then blastocyst. Blastocyst gets implanted over there, and later it forms ectoderm, endoderm, the three germ layers. Okay, the three germ layers, as in they are the three layers with which you get different organs being formed in your body, different organs, different things formed in your body. And then you can write this is the month-wise development plan in the case of human beings. 1 month this, 3 months this, and go on. Okay. Now instead of giving you 2 months, they can give you 2 months basically means 8 weeks. So you should be knowing at the end of 8 weeks, limbs and digits are being developed. Do not get confused with that. Okay. So please make sure you know about this. MCQ questions can come on the basis of this as well. Going ahead, we have draw sectional view of did I miss anything? No. Draw sectional view of seminiferous tubule of a human. Now, this is not something that we expect, but this is there in the question bank. Okay, seminiferous tubules have never been asked, but here is a question. So seminiferous tubule, I told you, if this is a testicular lobule, my dear children, inside that you have so many seminiferous tubules. When you cut it down and when you see through side it like this, okay, if something was like this, I cut it and I am showing you from here, this is how it looks. So these are the seminiferous tubules, my dear. What do you have over here? You will be having Sertoli cells, you will be having spermatogonia as well, and the spermatogonia will be dividing further to give spermatozoa. So inside it you will be finding spermatozoa. Okay. Now outside the seminiferous tubules you will be having interstitial space. This dark colored thing is interstitial space. You will be having interstitial cells. Now over here you have one more task to do. Label the following cells in the seminiferous tubules. So cells that divide by mitosis to increase their number. What are those cells which are going to divide by mitosis to increase their number, my dear children? It is spermatogonia. Spermatogonia. So how do you tackle them? So mark it over here. The innermost thing would be spermatogonia, and then say it's spermatogonia. Now cells that undergo meiosis 1. So spermatogonia, few of them will be specialized to form primary spermatocytes, and they are going to be the one that is going to undergo meiosis 1. So this answer is primary spermatocyte. It is just testing your knowledge but in a different format. So spermatocyte, just above it you can make spermatocyte. Okay, anything you can tag and write sperm, but it should be above, it should be going like that in inner side. Now cells that undergo meiosis 2, what are those cells? So primary spermatocyte divides and form secondary spermatocyte, which will be further going. So it is secondary spermatocyte that we have over here. Now cells that help in the process of spermiogenesis. Spermiogenesis, what are those cells that helps in the process of spermiogenesis? You can write Sertoli cells. You can write Sertoli cells over there. Sertoli cells, basically they release androgen binding protein, certain factors, right? So Sertoli cells can be your answer. Cells produced due to spermiogenesis. What do you get at the end due to spermiogenesis? The spermatids which are there are transformed into spermatozoa. That is the end result of spermiogenesis. So your answer becomes spermatozoa. That is the innermost thing that you see over here. Now this question says that help in the process of spermiogenesis. It's not just Sertoli cells. You can also mention Leydig cells, but make sure you're writing Sertoli cells first and then the Leydig cells. Okay, because Leydig cells releases the testosterone, right? It helps in the formation of testosterone, and Sertoli cells basically binds those testosterone with the help of the factors which are there. So that is kind of confusion which is there, but if you're writing the answer, please make sure you're writing Sertoli first, explain a bit about it, then Leydig. Explain. It's a 5 mark question, you can explain. Okay? For not losing your marks. Cool? Works? Chalo. Now answer the following. Given below is a flow chart showing ovarian changes during menstrual cycle. Okay, fill in the spaces with the hormonal factors responsible for the event shown. Okay. Okay, so what is the thing that is responsible for conversion of primary follicle to Graafian follicle? You have to tell about the hormones. What are the hormones? Primary to Graafian follicle, as in your follicles are maturing. So for follicle maturation, what do you need? Follicle stimulating hormone, my dear children, FSH. Okay, FSH majorly, and also a bit amount of LH as well. Now over here, Graafian follicle to basically ova. So here if I remove LH, I can write it over here. So for Graafian follicle to ova, the Graafian follicle ruptures. That's when you release basically the ova. That is what you mean by ova. It's secondary oocyte, I know that, but still ova is what they are referring to. Okay, so this will be because of LH surge, so I can write LH properly over here. So for this, let's not write LH, let's just write FSH. Here we can write LH. Now ova to corpus luteum. Ova, okay. What? Just a second. Fill in the space with the hormonal factors responsible for the. Okay, wait, just a second. Ova. Let me think about it. Okay, I'm taking a pause. Let me think about it. Okay, with the limited amount of knowledge that I have, I think they are trying to talk about progesterone over here, but it is actually released from corpus luteum. It's not like ova makes the corpus luteum. It's kind of confusing, but let's write progesterone over here. Okay, if at all I find some legible answers apart from this, I will be putting down in the comment section. But as of now, let's keep it like that. Works? It is the corpus luteum that releases progesterone, but somehow the arrows does not match. I think there is a typo error in that. Okay, now women experience two major events in their lifetime, one at menarche and the second at menopause. Mention the characteristics of both the events. What do you mean by menarche? Menarche is the start of the menstrual cycle, and menopause is the cessation of the menstrual cycle. So what are the changes that they find? So okay, they have mentioned. Okay, so the answer that I have written for myself was this only. It is luteinizing hormone that converts the ruptured follicle into corpus luteum, which secretes progesterone. So yes, you can also write your answer like this. I think it will be good. Yes, for this you can write FSH. For Graafian follicle to ovum, you can write LH and also estrogen. Okay, because it's estrogen surge and also LH, but majorly it is LH. Majorly it is LH. Okay, so please write it first, and then you can give an explanation. I write about it also, and then talking about this thing, that is basically once it is released, what is that? Something that maintains the corpus luteum. That if that is what the question is, then you have to write LH only, because LH is the one that maintains it. But please make sure you are writing that progesterone is released from corpus luteum. Will you be able to write that? Corpus luteum. Okay, make sure you write that. So this is why having an answer is really much important. These are the answers that even I only found out and right, but still there is confusion. That means yes, it is a confusion question as well. So make sure you're writing like this. As I said, for this, no, you can write that they secrete progesterone. Please make sure you're underlining this as well while you're writing. So your teacher knows that you know the answer, but the question itself is what? Kind of confusing in that sense. Okay? Works? And here also, please write luteinizing hormone, but also make sure that LH surge happens because of what? Estrogen. So here you can just write LH, and then later in your answer somewhere you can explain a bit about estrogen as well. Okay, now what are the changes that you find in menarche? It is the onset of menstrual cycle in the females, usually between 10 to 14 years. Now indicates the beginning of reproductive capability. Over here, permanent cessation of the menstrual cycle, usually between 45 to 50 years, and marks the end of reproductive phase with a decline in estrogen and progesterone level. And because of this hormonal changes, women face a lot of mood swings and things like that. So mention the characteristics. These are the characteristics that you can be writing about it. Okay, works? Chalo. Next, in the figure given below, part A and B show the level which influence the menstrual cycle. Study the figure and answer the questions that follows. So this is the same thing that we just studied in the menstrual cycle. So which hormones are represented in part A of the graph? I told you, this type of question comes. So A part is this, where FSH and LH is mentioned. Okay, so which hormones are represented? It is FSH and LH, and who is the one responsible for it? It is pituitary. Please remember, it is pituitary gland which releases it. Please don't write ovary and cum. Now which hormones are represented? Part B, my dear children, it is estrogen and progesterone. Who releases it? That is the question, and that would be what? Ovary is the one that releases estrogen and progesterone. Please remember, placenta releases after after fertilization and everything. So before fertilization, this is the thing that we have to remember. Now, role of progesterone and estrogen in the menstrual cycle. So as I told, as we discussed, this estrogen is secreted by the developing follicles. It promotes the proliferation of the endometrium in the proliferative phase, peaks before ovulation, and helps in LH surge, triggering what? Ovulation. Talking about progesterone, it is secreted by corpus luteum after ovulation, maintains the uterine lining in the secretory phase for possible implantation. If fertilization does not occur, progesterone level drops, leading to what? Menstruation. Works. Can you write that? Okay, please write that. Chalo. Now, we are coming to diagrams, and with that we are probably ending this particular chapter after a long time, right? So, draw a neatly labeled diagram of sectional view of seminiferous tubules. This is a diagram that you have to make if the question at all is asked to you. Now, draw a neatly labeled diagram of human sperm. So, over here, please remember you have acrosome at the top. This acrosome has enzymes to basically open up the plasma membrane and get into, what do you call it, the ovum, or say secondary oocyte cytoplasm, which is there. So, make sure you're right, knowing all these things, okay. Now, there are some enzymes. Now, going beyond that, you see this condensed region is the head region. Over here, you have the nucleus. The whole thing is the head, but just below the acrosome, you have the nucleus, okay. And just below that, you have a small region that is neck, and then you have middle piece which has so many mitochondria, and these mitochondria are going to help in the movement of the tail, my dear children. Works for you, okay. Okay. Now, one thing that I want to say is the whole sperm which is there, it is surrounded by a plasma membrane as well, which is mentioned right over here. Cool. So, please make sure you are drawing all this thing. Now, draw a neatly labeled diagram of sectional view of human male reproductive system. Now, this question was asked in the model question paper as well. Now, they asked the sectional view. According to your NCERT, the sectional view is this. The sectional view is this, because that's a sectional view, right? Now, this is a diagrammatic view that you have. This is the one that is usually we ask people to draw this. But if they're so sure about the sectional view, if they're asking that only, then we have to make this. If they have just mentioned, please put the diagrammatic view of male reproductive system, then this would be the answer for this. Okay, so please, I recommend both of you to practice both of them equally. Okay, if at all I get an information, I am trying to reach out to the dean. Okay, if at all I get an information regarding this, I will be informing you in the comment section below. So comment section is not just for you, but also for Jasmine ma'am. Okay, so yes. Now, next, draw a neatly labeled diagram of a sectional view of the human female reproductive system, a star question from this particular chapter. Very, very important question. So, this is usually being asked. Make sure you're thoroughly known with the diagrams. Okay, you have the uterus, 3 layers of the uterus. You have the ovary, the fimbriae, the infundibulum, the ampulla, the isthmus, the uterine fundus, which is there, the cervix, the cervical canal, the vagina, all of it. Please make sure you're mentioning it properly. Next, draw a neatly labeled diagram of sectional view of mammary gland. This is also a question from the model question paper. So, if you know, in model question paper, certain questions have come in, and one such question is about the mammary gland. So, this is the mammary gland, my dear children. If you know about the labelings, you would be able to draw it also properly. So, first thing, you have a lot of fat in there. So, this is the fat that is there. Inside that, if you go, my dear children, you have 15 to 20 mammary lobes. Okay, so these things that you see over here, can I zoom it in? Yes, ma'am. So, these lobes, this is one lobe and 2 lobes. You see there are different lobes over here. So, these lobes that you see, they are mammary lobes. You have 15 to 20 of them. Now, inside each mammary lobe, you would be having so many alveoli. You will be having so many alveoli, mammary alveoli. Now, alveoli will be opening up into tubules. These branches that you see, 1, 2, 3, 4, 5, all these branches are tubules. Tubules will be opening into what? The ampulla. Okay, this thing is ampulla. So, we have mammary lobes, then alveoli, then tubules, then ampulla over here. This ampulla which is there, oh not ampulla, sorry, the duct which is there, not ampulla, duct. Okay, first comes duct and then comes the broader region which is known as ampulla. From the ampulla, all of them are going to join to form lactiferous duct, and then this is going to go out through the nipple or the areola. Got it? Fine? Fine. And with that, we complete human reproduction. Give a big tap on your back. You guys did it. My dear children, if you are with me solving a lot of questions up here, I'm telling you, you're going to take away a lot of information which would help you solve a lot of MCQs as well. Trust me on this, and it's not just for increasing views. As I said, this video is kind of too long. We have to speak for too long. Still, if we are making it, then understand there's a purpose added up for this particular video. And if you're liking it, do not forget to like and also comment down, because we also need to get appreciated, right? Who will appreciate it? Appreciate us apart from you guys. So yes, please do that part, and let's begin with the next thing that we have, that is reproductive health. So Over here, let's start with the first question. Mention the major task under RCH program which is there. RCH, the full form of it is being asked. What do you mean by RCH? Reproductive Child Health Care. So you feel like it's RCHC. Reproductive Child Health Care is also there. Please make sure health care is treat it as one word, okay, even though it's not, but yeah. So Reproductive Child Health Care program, that is a thing, and the major task was to, for what? Look after the maternal and child health care, my dear children. So it includes the maternal care, infant, immunization, nutrition, family planning and everything. And apart from that, reproductive health services to improve that. So example, it covers contraception, STD prevention, infertility treatment, safe abortion, and also adolescent health education. So these were the two major tasks under RCH program. Now these are the basic things, but we know what? We are not able to make up the basic things. We know the big things, we can make up that, but not the basic things. So make sure you're knowing this. First is maternal and child health care. Second is to improve what? The reproductive health services which are there. 2 mark question, you get 2 points for this and you get marks for it. Now which days of menstrual cycle are considered fertile period and why? Fertile period as in, during this period, if two people have sex, okay, there are chances that it can lead to fertilization and then later a baby there. Okay, so this period is known as fertile period. Why do you call it fertile period? Because in this period only there are chances that ovulation can happen, the ovum can be released, and the sperm, if it comes in the same time, both of them, they will fertilizing. You get it? So that is why it is known as fertile period. So day identify. The fertile period is usually between day 10 to day 17 of a 28-day menstrual cycle. So day 10 to day 17 is known as fertile period. Anytime ovulation can happen. It usually happens on the 14th day, but for some people obviously it can change here and there. Okay. Now the reason the period is considered fertile because the ovulation occurs around day 14 when a mature egg is released. Sperm can survive for 3 to 5 days. Do you know that? Once copulation has happened, the sperm can actually survive for 3 to 5 days inside the human body. And suppose the egg is being released during that particular day, then what will happen? Basically fertilization can happen. So before ovulation, after ovulation also, it's not safe for the sperm to be released. So that's why the fertile period you should not be having sexual intercourse. And the egg remains viable for 24 hours. So egg can remain viable only for one whole day, that is 24 hours. Within this, if the sperms are still there, fertilization can happen. Now this is most likely for conception if unprotected intercourse occurs. It is the most likely time for what? Conception, for conceiving a baby. Okay, I hope you got the answer. Now getting ahead with the next question. Parents of a 1-year-old child wanted to space their second child. The doctor suggested CUT. Explain its contraceptive action. What do you mean by CUT? CUT stands for copper T. Okay, and I'll tell you one thing about copper T, or basically these are, whenever you're saying the doctor is suggesting this to take a contraceptive action like CUT, CUT is an intrauterine device. Intrauterine device, that means the doctor is going to put this device into the uterus, inside the uterus. And what does the CUT do? CUT will be releasing, CUT will be releasing what? Copper ions. And these copper ions are going to hold the sperms like this. Hey, sperm, you cannot move. You cannot move. You should not be moving. So the sperm's motility is being reduced. Do you think it can go and fertilize the ovum over there? No. Okay, and one more thing, it can also lead to increase in phagocytosis of sperm. That means the sperms can be killed also inside. So it is a contraceptive method. We do not want fertilization to happen, so obviously let's kill the sperm. Get it? And also it creates a very unfavorable environment for implantation to happen. Even if fertilization has happened, implantation is not going to be possible over there. So these are the things that you have to remember. So it increases phagocytosis of sperms. The presence of CUT enhances the destruction of sperms by white blood cells in the uterus. Okay, but I'll recommend you to write this point first. This is the most legible point, that is the second point. Reduces sperm motility and fertilizing capacity. Copper ions released by copper T suppresses the sperm movement and reduces their ability to fertilize the egg. This is more associated to the another intrauterine device. Okay, in IUD you have 3 types. One is non-medicated, one is copper releasing, and one is medicated. Non-medicated one do not release any hormones. Okay, nothing. It just stays there as a foreign particle, and whenever something foreign gets inside our body, what will happen? Our body will react to it. It will produce white blood cells, and these white blood cells, obviously when it is released, any other cell coming inside the body would be killed by the white blood cells. So that is how non-medicated IUDs work, and this point actually suits non-medicated IUDs better. So I'll just cut off this point. Okay, second point I have, it reduces sperm motility. This is better, and it prevents implantation. CUT creates one unfavorable environment in the uterus, preventing the fertilized egg from attaching to the uterine wall. This is also fine, but make sure you're writing this point first. Apart from CUT, do you have anything else? Yes, CU7, okay, and then you have multi-load 375. Multi-load 375 is also a drug like that. Now, going ahead, why do women use Saheli, the oral contraceptive pill, as a well accepted method? Why do women use Saheli, my dear children? Saheli was actually made by Central Drug of Research Institute, okay, CDRI, Lucknow. And why was it widely accepted? It's still widely accepted. Why? Because it is a non-steroidal pill, my dear children. It does not release any hormones. So usually the oral pills and all females have, it's going to release certain hormones, okay, and obviously this creates a hormonic imbalance. But in the case of Saheli, it is not going to release any hormone. It is a non-steroidal oral contraceptive that you have it, okay. And how do they work? Basically, estrogen, right? And estrogen is very important for endometrium maintenance and things like that. Now the receptors which are there for estrogen, on which the estrogen comes and act, and then it helps in the formation of endometrium, these receptors are blocked. These receptors are modified in some or the other way. So the estrogen working is not happening properly in them. So estrogen working would not be happening properly. So it works as a non-steroidal hormone by modifying the estrogen receptors, my dear children, and not allowing ovulation as well as what? Implantation. So both ovulation and if estrogen is not there, do you think ovulation can happen? Estrogen is the one that says LH to increase its amount. No. So if estrogen is not there, no ovulation, no implantation. Now it is convenient and safe. It is taken once a week. It has minimal side effects and it is non-hormonal and is easily reversible for future pregnancy planning. So it's a once in a week pill, minimal side effects, non-hormonal and easily reversible as well. Okay, can I remember it? Once in a week, minimal side effects. Rest all the oral pills, please remember this. Rest all the oral pills have to be taken 21 days, except the days of your menstruation, that is from almost 5 to 7 days, you can give a gap. And after that, each and every day you have to take the pill, and it releases a lot of hormones in our body, which obviously we do not want. So this is a once a week pill with minimal side effects, non-hormonal, very important, and easily reversible as a non-steroidal thing. Got it? Going ahead, list one of the contraceptive method, each in male and female, with poor reversibility, as in the reversibility is very poor. If you want to reverse it, you cannot. For example, for copper T and all, we can remove that from the uterus. Okay, so it is reversible. But over here, there is something that is not reversible. It is the surgical method. It is the surgical method. So I told about vas deferens. Vas deferens is the one that carries the sperm. What if we cut? What if we cut the vas deferens? This is known as vasectomy. Now, vasectomy, when it is done, will the sperm still be transported? No, the sperm is not transported. Hence, it is a reliable method of contraception, my dear children. Is the sperm made over here, my dear children? That is the next question. Again, a very important question. Are the sperms being made over here in the testis? Yes. So please do not go and write spermatogenesis does not happen in vasectomy. Spermatogenesis happens. Okay, sperms will still be formed, but it cannot move out. It cannot move out. So whatever sperms reaches over here, what will happen to that, ma'am? They will die. Okay, they will all degenerate. Okay, please remember that. Now you have vasectomy. After that, you have tubectomy. What happens in tubectomy? There is a fallopian tube. The fallopian tube is the one that carries the ovum. Again, the same thing happens here. Also, the fallopian tube is cut and tied. Now, will the ovum move there? Can the sperm come over here? No, there is no chance to meet. Even though it's a sad story, but it's a very useful method. Okay, if you do not want children later on. So it is one very important contraceptive method, but with poor reversibility. That is the only drawback that it has. So for example, suppose as of now I'm thinking that I don't have kids. I don't want to have kids. Then I did tubectomy. Then later I realized, oh shit, I wanted kids. Then what? So that is the thing. Okay. Now going ahead. So reason for yes. So vasectomy is a surgical method where the vas deferens is cut and tied, preventing sperm transport. Here a surgical procedure where the fallopian tubes are cut and sealed, blocking the passage of eggs. Now reason for poor reversibility is that both methods involve permanent surgical intervention, making natural fertility restoration a very challenging process. That is why we say it is having poor reversibility. Works? Going ahead with the next question. Okay, over here also one more question. Does menstruation have a dear children? Does menstruation happen over here? Yes. Even if a person has undergone tubectomy, menstruation still will be happening because ovum will be anyway released. Every time our body will feel, our body does not know there is a cut has been made. You get it? So it does not know how to react. So anyway, every time it will make up the endometrium and everything. It will happen naturally. But the only thing is that the ovum is never gonna make it. It's just like that. Fertilization is not happening. Okay. Now, shown below is a surgical method. This type of question can come. This is why I added. I have not put any number for this because this is an extended version of the question. Now, give the technical term for the process shown above. So, you should know what is it. It is what? vasectomy. What is the principle behind it as a contraceptive method? It does not allow transport of sperms. How is the process carried out? So how is the process carried out? There's a small incision made in the scrotal sac, and slowly everything is being cut with the help of that. Cool? Now, mention 4 early symptoms of STD that occur in the genital region. Again, a very basic thing that we tend to not even put our attention into. Okay, but still, as I said, you should be paying attention to small details. So there are some early symptoms of STD, sexually transmitted diseases, that can occur in the genital regions, my dear children. First is itching and irritation. So persistent itching or discomfort in the genital areas is one thing. Second thing, painful urination. While you urinate, it's kind of painful. There is a burning sensation or pain while urinating. Third thing, unusual discharge. Unusual discharge as an abnormal fluid or pus-like discharge coming from the genitals. That is also thing for STD. Now, sores or ulcers being found, appearance of blisters, sores or warts around the genital area, this is also a symptom. So itching and irritation, painful urination, unusual discharge, and sores and ulcers. Okay, even while reading it I feel bad. I don't know for the people, how do they feel it? Oh my God, it's sad, right? So you should make sure, children, please be away from all the sexually transmitted diseases. Please do not have multiple partners. I am being very open over here because I know it's your age to experiment a lot of things, but please do not do that. You do not know where you're going to land up to. Okay, in what situation you're going to land up to. So please make sure, all my dear children, please just focus on studying and enjoying your life as much as you can, apart from doing all these things over there. Okay, I am here talking to you as my friend. Chalo. Because you listen to what your friend says, yeah. So suggestion is 2 arts that can help the couple to have a child where the problem is with the female partner. What do you mean by art? Assisted reproductive technologies, as in your body cannot do it by itself, so somehow someone is assisting you. Okay, so there are 2 methods. You're asked about 2 methods where a couple is not having child and the problem is with the female partner. So in female partner, first thing you have to think of the problems that can be caused. Either she is not producing enough amount of egg, or the egg she is producing is not great. Second thing, uterus is not very favorable for implantation. Every time fertilization happens, but implantation is not happening. So these are the 2 scenarios that we have. Okay, or somehow in the body itself fertilization is not happening. 3 scenarios. So I'll recommend method 1, zygote intrafallopian transfer. Okay, when fertilization is not happening naturally inside the body of the female. So what do? We take her egg and we take her egg and also the sperm of the person which is involved. We fertilize it in the laboratory condition, and then we put this particular embryo, zygote, which is going to form. Zygote, or basically if it has divided to form an embryo, if the embryo is in 8 to 16 blastomere stage, that means it has 8 to 16 cells only. Okay, then you can put it directly into the fallopian tube. Directly into the fallopian tube, the rest of the formation will be happening inside that, inside the body. Second thing, if you think that the you need a direct implantation, the embryo has divided more than 16 cell stage, then you have to put it in the uterus. So for that you do intrauterine transfer. Inside the uterus you transfer it directly, not in the fallopian tube. If you put it in the fallopian tube, it will lead to ectopic pregnancy. That means pregnancy at other spots apart from the uterus. So please don't do that. Okay, if it is only up to 16 cells, then you can put it in the fallopian tube. Apart from that, please make sure you're putting inside the uterus. Now, here you need direct implantation, usually in the case of blocked or damaged fallopian tube. Yeah, if a person has blocked or damaged the fallopian tubes, then you can put it inside directly the uterus. So that is also one problem, right? Now, the third problem, as I said, if the female is not producing viable gametes, that is a big problem. So you do gamete intrafallopian transfer, where what you're going to do is you are going to take a donor's egg. Okay, donors is someone else's egg, and you're going to put it, because the female is not able to produce egg by himself. So used when the female is not producing viable gamete, but allow fertilization to occur naturally in the fallopian tube. So she is allowing fertilization, everything to happen, but she is not able to produce a viable amount of gametes which are there. So you insert the gamete into the fallopian tube, but this gamete is going to come from another donor woman, and then you let normal sexual intercourse to happen, because rest of the things can happen normally in her body. So these are the 3 scenarios that we have. Please make sure you're writing it properly. One is where I'm saying that fertilization is not occurring naturally, so you're doing it outside. IVF you're doing, okay, where you're going to introduce the zygote or the embryo, which is up to 8 to 16 cell blastomere stage. You can put it in the fallopian tube. Suppose the fallopian tube itself is damaged and fertilization is still not happening, you're going to put the embryo, which is more than a 16 cell stage, into directly the uterus. This is known as intrauterine transfer. Then you have the woman who is not producing what? The gametes itself. So in that case, the female is not producing viable gametes. So another donor female gives it, and then you put it in the fallopian tube. Their fertilization can happen naturally. Okay, works. Now suppose the uterus itself is not great for implantation. You do something that is known as surrogacy. You do something that is known as surrogacy, where basically the gametes can be taken from the mother and the father. They can be fused, and then the zygote which is formed, the embryo which is formed, can be put in another female's body. Okay, that is known as surrogacy. Now, explain the technique that can help a healthy married woman who is unable to produce viable ova, but can provide a suitable environment for fertilization and further develop and wants to bear a child. Again, she is unable to produce a viable ova. Okay. First thing that comes into your mind is what? Gamete intrafallopian transfer. You can write that first. I didn't write it as first, but you can write it as first. Please write gamete intrafallopian transfer as first point. If the female is not able to produce the gametes, but fertilization is not occurring, okay, is not able to produce the gamete, but fertilization is not, okay, this is okay. If the female is not able to produce the gametes, donor eggs and the husband's sperms are directly placed in the fallopian tube, allowing natural fertilization inside the body. It's not, you don't need husband's sperm also as of now. Okay, you just put donor eggs inside what? Are directly placed in the fallopian tube, allowing natural fertilization inside the body. You get it? So what do? Female is not able to produce the egg, the ova. Okay, but can provide what? A suitable environment. So you take the donor eggs, put it in the fallopian tube of her body, and then natural intercourse can happen, fertilization can happen naturally. Now, this and this can also be done. Zygote intrafallopian transfer. For that you have to do IVF first. What do is that she is not able to produce viable ova, right? So we take donor's egg and we fuse it with the male partner's egg in the laboratory condition. Then the zygote and embryo that you get, you can put it back into her body because she has all the reasons for implantation. Okay, she does not have any problem with implantation. Now this is intrauterine transfer, where again the embryo which has divided more than 16 cells, then probably you can directly put it in the uterus as well. Again, this embryo which is being formed, it is from the donor's egg and the husband's sperm. Okay, so IVF can also be done over here, and this can also be done over here. So IVF means in vitro fertilization. Inside the laboratory, fertilization is done, and then post that you can do all this thing. First you write this, and then you can go ahead with this. Make sure you are writing donor's egg. If you are writing something else and coming, you are not gonna get marks. You're gonna lose your marks over there. Okay, she is not able to produce egg, so please do not write mother's egg. It is donor's egg that is being taken up. And please write this first because this is the actual answer for this question. Okay, now a large number of couples all over the world are infertile. Such infertile couples could be assisted to have a child, okay, could be assisted to have children through certain special techniques commonly called as assisted reproductive technologies. Give technical terms for each of the method described below. Transfer of ova collected from a donor into the fallopian tube of another female who cannot produce functional ova. It is gift. She is given the gift, gamete intrafallopian transfer. Okay, semen collected from the husband or a healthy donor is artificially introduced into the uterus of a female, into the uterus of a woman. So, semen which is collected from another person. When is this being done, my dear children? When is this being done? Basically, when the man, okay, might have some arousal issues, okay, erectile dysfunction issues, or probably he might not be producing too much of sperms together. Okay, a lot of things can happen. So basically at this point of time, artificial insemination is done, where basically you take the sperms from the guy who is there and then you put it inside the uterus of the woman, because probably erectile dysfunction or anything could be there. You can also do it with the donor sperm also, if at all it is needed. Okay. So semen collected from a husband or a healthy donor is artificially introduced into the uterus of a woman. It is artificial insemination. Now, an embryo is formed in the laboratory by directly injecting the sperm into the ovum. This is known as IVF, in vitro fertilization. So as I said, in the laboratory, that means in vitro fertilization. In vivo means inside the body. In vitro means outside the body, that is in the laboratory condition. Is it clear? Will you be able to answer all these questions, my dear children? Do you think your confidence is getting boosted up, or do you feel that, ma'am, I don't know any answer? Baba, if you do not know any answer, this is why I am here. I am explaining you things for your own good. So please do not feel that, okay? You are gonna ace it. You're gonna ace the exam. You're gonna feel so good. Does not mean that everything is going to go away. No, everything is going to stay intact in your head. Okay. Now, one very important thing that I want to tell is that, yeah, so in the 2 marker, 3 marker, or 5 marker, 2 marker, 3 marker, I think in that particular video, I have given so many other questions also which have come in previous years. So these questions, as I am telling you, all of them are from question bank, are from the question bank that the government gave. Now, I solved the previous year questions already. I solved the model paper also. Please go watch it. Please go watch all those videos. I have given you a timetable also. Please follow it. It will be really, really, really helpful for you. Okay? Will you do that? Yes. So in that, the question is, what is infertility? What are the reasons for infertility? So the physical, psychological, and all those reasons. Please make sure you're writing that. Drug use and all. Now, how is amniocentesis performed? Why is it legally banned in our country? What do you mean by amniocentesis? You know what? So this is the uterus. Inside the uterus, the cute little baby is gonna lie over here. And you know what? It looks like a teddy bear. It is covered by an amniotic sac. An amniotic sac, you have amniotic fluid. And what happens is that certain cells of this baby is gonna shed down over here. Okay, it's gonna shed down over here. Now you're gonna take, in amniocentesis, you're gonna put the injection, you're gonna take this fluid. When you take this fluid, you get the cells also. In the cells, what do you have? You have the DNA and everything up there. Agreed? Agreed? So you usually take this for understanding if the child would be having some genetic abnormalities such as hemophilia, such as sickle cell anemia, and such as Down syndrome, some genetic disorders. You get it? If at all it's there. But people have started using this for something else, for sex determination, because you're talking about chromosomes and there are sex chromosomes. If you determine the sex chromosome, you can know the sex of the child. And if it is a female child, then the people were starting to do what? Female feticide, which is why it was legally banned in our country. Okay? So what is it? The first part of the question is, how is amniocentesis performed? So a thin needle is inserted into the amniotic sac using an ultrasound guidance, with the help of ultrasound. Otherwise you're going to directly pinch the baby. No, you don't want to do that. Now a small amount of amniotic fluid is extracted. The fluid contains the embryonic cells and is tested for genetic disorders and chromosomal abnormalities such as Down syndrome, hemophilia, and sickle cell anemia. Why is it banned in India? Because it is misused for sex determination, leading to what? Female feticides. Females are killed, which is known as female feticides. Works? Chalo. Next thing. Mention the parameters that indicate improved reproductive health of the society. Parameters indicating reproductive health is what? Decline in maternal mortality rate, that means mothers are dying less. Okay. Decline in infant mortality rate, that means babies are dying less. Increased life expectancy, better healthcare leads to what? Longer lifespan. Higher awareness of contraception, people are aware of it. That means this society is reproductive healthy, right? They won't be making so many kids up there because you do not have resources to maintain them. Now, reduce cases of what? STDs, sexually transmitted diseases, better maternal and child care, balanced sex ratio. All of these things are again the parameters that decides what improved reproductive health of a society. Okay? Okay, chalo. What are contraceptives? Mention the qualities of ideal contraceptive. Contraceptive is something that would prevent what? Conception. That would prevent pregnancy. Those are contraceptive. So contraceptives are methods or devices used to prevent pregnancy by interfering with fertilization, implantation, or ovulation. Fertilization, baby going settle down, that is implantation, or ovulation as in release of ovum. Okay, what are the qualities of the ideal contraceptive? First thing, it should be highly effective, obviously, it's not failed, right? It should be reversible. It should be reversible, even though surgical methods are not, but still, we'll say we are putting on a contraceptive because they are contraceptive, but they have poor reversibility. Okay, now use a condom. Condom can be removed. It is highly reversible. Now there should be no side effects, obviously. It should be easily available for all and it should be affordable, otherwise people cannot afford it and there will be obviously a lot of human beings being created. Now it should be user friendly. Okay, you should not always need someone else's help to take use of it, otherwise people are there who feel shy and things like that. And then it should not interfere with the sexual desires of human beings. It is just a mode of contraception. It should just prevent pregnancy. It should not prevent what? Having the desires. So it is highly effective, reversible with no side effects, easily available, user friendly, should not interfere with the sexual desires or other features of contraceptive methods which are available. Works? Chalo. Categorize the following into different kinds of IUDs, such as non-medicated, copper releasing and hormone releasing. So I told you we have 3 types of IUDs. Okay, IUDs are widely accepted, most accepted thing. Please remember, IUDs are example of the most accepted contraceptive. This is question that comes. IUD is the best for spacing children. If you want a child after 5 years, so basically use an IUD. Okay, so 3 types of IUDs there: non-medicated, copper releasing and hormone releasing. In non-medicated comes lipase loop. What is the principle of it? Basically putting just something inside the uterus which is not going to obviously harm the uterus very much. But when something gets inside the uterus, what dear children, our body feels it's very foreign and our body will react to it. It releases WBCs. Now when the sperm comes in, it will feel that it is foreign, it will kill the sperms also. So it will lead to phagocytosis of sperms which are there. Now copper releasing IUDs, we already discussed about that. There is CUT, there is CU7 and multi-load 375. They will be releasing copper and it messes up with the motility of the sperm and also does not allow implantation to happen. Okay, hormone releasing IUD, that is progesterone and LNG20, my dear children. They are going to release the hormone that is progesterone. Okay, and when it is being released, what is happening? Usually progesterone helps in all kind of good things, that is implantation, endometrium maintaining and everything. But when their level is way too much, they are not going to do the same thing. Okay, that does not allow the ovulation to happen. So I'll say ovulation is not happening. Second thing, they do not allow the endometrium line to thicken. Endometrial line lining which is there, it does not allow it to thicken, it's thin. So in thin situation, do you think implantation can happen? No. Now cervical mucus which is there, cervical mucus thickens, my dear children. What happens? You know, the entry of the uterus is cervix region. Here if there is a mucus plug being formed, do you think the sperm can get entry to it? No. So all of this thing has been done by these guys. Okay, progesterone and LNG20. And the oral pills also, the oral pills which are being used as a contraceptive methods, my dear children, as I told you, 21 days it has to be taken. It is also a combination of progesterone and estrogen, or basically just progesterone. What it does the same thing. It does not allow ovulation to happen. It does not allow the maintenance of endometrium. Okay, it creates a cervical plug, mucus plug which is there. It does not allow the entry of sperms and all the things. Please make sure you know it. Okay. Now, at the time of independence, the population of India was 350 million, which exploded to over 1 billion by May 2000. List any 3 reasons for this rise in population and any 2 steps taken by the government to check this population explosion. So, reasons why it increased is decline in death rate, increase in the birth rate, and better healthcare and nutrition facilities were provided. Now, steps, obviously, government cannot go kill people. Okay, government cannot reduce the efficiency of healthcare, because it is all for betterment of the human people, right? So what are steps taken up by the government? Basically, they started up having family planning programs, promotion of contraceptives, sterilization methods, awareness campaigns and everything. Now the legal age of marriage was also increased. Minimum age of marriage for a girl is 18, for a male is 21, and this all led to delay to childbirth. Otherwise they were just producing children, you get it? So yes, these are the steps that were being taken by the government, and these are the reason for the population rise. Now, define venereal disease. Mention the modes of transmission of HIV, hepatitis B virus. Define venereal disease. How do you define venereal disease, my dear children? So, basically, they are also known as sexually transmitted diseases and are infections spread through sexual contact caused by bacteria, viruses and parasites. Okay, venereal diseases means sexually transmitted diseases only. They are the infections which are spread through sexual contact caused by bacteria, viruses and parasites. The mode of transmission in the case of HIV and hepatitis B is being asked. So unprotected sexual contact, first thing, because STDs are transferred through body fluids. By body fluids, I mean genital fluids. By body fluids, I mean blood. Okay, so it is infected through the semen, the vaginal fluids or the rectal fluids which are there. Second thing, blood transfusion, receiving infected blood or blood products. That also HIV and things. So what do you say? Human beings who have some diseases where you need repeated blood transfusion, you have to be really careful what type of blood is being inserted in the body. Sharing needles and syringes. Again, when you insert syringes and needles, same. Okay, if you share, the blood is in contact there and you're putting the same thing over into your body, so that is a big problem. So using contaminated needles is a thing. From mother to child also it happens during pregnancy, childbirth or breastfeeding, if at all. For breastfeeding, it is still a big debate that is going on, okay, but still for other things such as childbirth and pregnancy, during that point of time anyway, the blood can mix and match, and that can lead to what? The disease which is there. Works? So this is about HIV and hepatitis. That means you're talking about the sexually transmitted diseases, and please remember 3 diseases which are not going to be cured. They are non-curable sexually transmitted diseases. One is HIV, second is genital herpes, and the third one is hepatitis B. So HIV starts with H, hepatitis B starts with an H, and genital herpes starts with G, but yeah, H is there. Herpes. Okay. Going ahead, the picture shows the contraceptive device used by women. Identify the contraceptive device shown in the picture. What is this thing, my dear children? These are implants. These are implants. What does it contain, my dear children? It contains progesterone or progesterone estrogen combination. How is it used? Basically, put it under the skin. You put it under the skin. Not you, doctors will do that. It is inserted under the skin of the upper arm. Now, how does it function as a contraceptive? Basically, again, the hormones are being released, so it prevents ovulation, thickens the cervical mucus, alters the uterine lining. Whenever the hormones are there, just understand these are the points that you have to write. Works. So I hope you will recognize this thing. Now, the 2 questions that I have put just for you to know that these type of questions can also come. Okay, it has come in the previous years. So mention the 2 medical grounds on which pregnancy are subjected to termination. For the 2 medical grounds, so first thing, there's an act, Medical Termination of Pregnancy Act, according to which whenever it is risk to mother's health, okay, if continuing the pregnancy possesses a risk to the physical or mental health of the mother, at that point it happens, or risk of fetal abnormality. If there is a substantial risk that the fetus may be born with physical or mental abnormalities at this point of time, also basically MTV is totally legal. Okay, fine. Or basically if it is rape or any situation like that at that point itself. Okay. Next thing, what are emergency contraceptive? Right there, hormonal combination. So emergency contraceptive, again, they are the pills that are used to prevent pregnancy after unprotected sexual intercourse. Now, they have to be taken usually within 72 hours after the event. Okay, administration of progesterone or progesterone estrogen combination or IUDs is going to be acting like emergency contraceptives. The hormones create all the things. Even if ovulation has happened, even if fertilization has happened, implantation would not be happening with the help of this. Okay. And with that, we complete the chapter. We're getting forward the next chapter, that is evolution. Are you ready? I hope you're ready. Just take a 2-minute break and come back really, really fast. Okay? Okay, let's get into it. Yeah, let's complete evolution as well. Starting with the first question, write a short note on Big Bang Theory. What is Big Bang Theory, my dear children? So listen. We believe that there was a big bang that happened, there was a huge explosion that happened, which is why we got the primitive earth and all the small things that are there. The whole galaxy was formed like that. And at this point of time, my dear children, there were 2 gases which played a very important role. One was helium and the second one was hydrogen. Both these gases were there, and these gases, okay, and at this situation when this huge explosion happened, the temperature was really high and there was a reducing environment. That means there was no oxygen. Temperature was really high and there was a reducing environment and there was presence of helium and hydrogen. Now slowly what happened, because of gravity, the hydrogen and helium settled down all over these things. Slowly these guys slowly turned into what? Molten masses. Suppose this is the earth we are talking about. The earth turned into molten mass and started releasing certain gases, my dear children, such as, please remember the names, methane, CH3 or CH4, okay, methane, and then you have ammonia. Okay, and then you had ammonia, and water vapor was there. Okay, and then yes, carbon dioxide, everything was there. Yes, yeah, carbon dioxide was also there. Fine, these were all being released over here. Now the sun is also there. The sun is going to act on the water vapor and is going to break down the water vapor into what? H and O, my dear children. This O can basically combine with methane, ammonia and things like that to give rise to what? Organic compounds, which you see. The organic things, these are inorganic things, and then can give rise to organic things. From these organic things, when they unite, for example, amino acid came together to form proteins. Nucleic acid came together to form what? RNA and everything. All of this came together to give rise to what? The first life forms which are there. And obviously you need oxygen. Where do you get the oxygen from? From the water vapor. When the water vapor was getting broken down, my dear children, you get oxygen and hydrogen. This oxygen obviously can go react with other oxygen to give rise to what? O2 and things like that. Suppose there is O2, this O2 can go react with other O to give O3, and slowly what happened? Ozone was formed. O3 means ozone, and slowly our earth was covered by some layer that is known as ozone, protecting it from the harmful UV radiation which was there. So this is what you mean by Big Bang theory. Okay, there was a huge explosion. The universe began with a massive explosion, Big Bang, leading to expansion and cooling, formation of elements and galaxies. So hydrogen and helium formed first, later condensing under gravity to form what? Galaxies. Earth's formation. The Earth formed in the Milky Way galaxy from the gas and the dust which was there. Early atmosphere and oceans, gases like water vapor, methane, CO2 and ammonia were present. Cooling of them. Okay, later what happened? The temperature came down. Initially temperature was way too much, temperature came down. When the temperature came down, all this water and everything settled down into the what do you call it, the free spaces which were there, forming what? The oceans and everything. Now origin of life, it appeared around 500 million years after the Earth formation. So Earth was formed 4.5 million years ago. So after that, this was been done. Was, will you be able to write this? Easy, no? So this is in the question bank, this is nowhere in the model question paper, or it was not asked in the previous question papers as well, previous year question paper, but still we can expect. Now, how could the experiment of Louis Pasteur dismiss theory of spontaneous generation of life, my dear children? So, first thing, what do you mean by spontaneous generation of life? Suppose there was a sack of things being kept, cereals, rice, okay, being kept. And one day I noticed that there is a small chutku there, there is a small mice over there. And I say, oh my God, from this sack of rice which is there, or wheat which is there, a new mice is arising. This is theory of spontaneous generation. Okay, the things basically just come from there. Life could rise spontaneously from non-living matters which is there, my dear children. But then Louis Pasteur got really angry. How can they even think like that? Children. So basically he did a swan neck flask experiment, my dear children, where what he did was he had a flask like this, a beautiful pre-lit flask. He had a broth over here, he sterilized the whole flask and everything, and he noticed that there was no growth of anything happening in this particular broth. Then what he did was, he did some changes to the flask, he kept it like this. There was no swan neck over here. The broth was still there, and still then he noticed there was growth of certain things over here. Then he realized probably over here, even when things were coming, they were getting trapped, because there's a loop like this. So they were getting trapped, which is why they are not able to enter. And that is why there was no life forms observed over here. This was also sterilized, but here the life forms appeared because there is no one to hinder, the things can get in and give rise to new life forms. So, first thing, theory of spontaneous generation says it was believed that life could arise spontaneously from non-living matter. Example, maggots from decaying meat, microbes from broth and things like that. Pasteur's swan neck flask experiment said that he boiled nutrient broth in a swan neck flask to kill microbes. Flask remained sterile as long as the neck was intact. When the neck was broken, the microbes entered and the broth showed what? Microbial growth. Conclusion, life arises from pre-existing life, that is biogenesis. This is known as biogenesis. Life that is there here, it is existing from pre-existing life, my dear children. It is not occurring spontaneously. Disapproved the spontaneous theory generation theory, confirming that microbes come from the air and not from the non-living matter. But he didn't tell, where did that life come in the air? What was responsible for that? For this, the next theory came, which was chemical theory, which was chemical evolution theory, my dear children. So even Louis Pasteur could not explain that. He just said the life came from pre-existing life. Cool? Now, I think the next question is that, next question is about chemical evolution. What do you mean by chemical evolution? So basically, children, we know that according to Big Bang theory, if we go, there are certain gases, that is methane, ammonium, carbon dioxide, hydrogen, helium and things like that. Temperature was really high, and basically the reducing atmosphere was there was no oxygen up there. So at this point of time, we believe that because of these conditions which were maintained, the gases were reacting among themselves. These inorganic things which were there, these inorganic gases which were there, they combined to form organic compounds, such as what? Amino acids and proteins and everything like that. Okay. Now, these organic compounds, when they were giving rise to life forms, you call this as chemical evolution. You call this as chemical evolution. Who put this? Oparin and Haldane said this. They suggested that life originated from simple inorganic molecules which gradually form complex organic compounds under Earth's condition. Life originated from what? Inorganic things, okay, simple inorganic molecules which are going to combine together to form complex organic molecules under Earth's conditions. Now, Miller's experiment. Stanley Miller tested chemical evolution by stimulating early Earth's atmosphere, that is methane, ammonia, hydrogen and water, in a closed system. Okay, to prove this, Miller did the experiment. This was the experiment about. So he had a closed system. Over here, as you can see, there was water. You would be asked to make this diagram, very important diagram from this particular chapter. Very, very, very important diagram. Okay, so here water is there. From here, the water, the vapor are going to come up. Over here we have putting electrodes for creating a high temperature, and this is a closed setup, so there is no oxygen as well. At this particular point of time, what do you observe is that there will be a lot of gases in there. Okay, there will be a lot of gases in there, and they're going to combine. They're going to combine to form something. Where do they form something? Over here, if you find the water, there are so much organic compounds. Where did they come from? From all these things. And this was the experimental proof for apparent Haldane's thing. Who did it? S.L. Miller. Okay. So he tested chemical evolution by stimulating what? Early Earth's atmosphere by doing all this. Applied electric spark lighting to initiate what reactions? Findings. After a few days, organic molecules like amino acids or building blocks of life were formed. Provided experimental proof that life's basic components could form spontaneously from inorganic molecules, supporting what? Chemical evolution theory. Life, organic things that you see, my dear children, are going to come from the inorganic things which are there, and this is what chemical evolution talks about. I hope you got that. Chalo, next thing. Same type of questions can be coming for the same thing that is there. What were the different gases used by S.L. Miller in his experimental setup? What were the different gases? Methane, ammonia, and then you have carbon dioxide or hydrogen, and helium and everything. Water vapor is also important. Okay, it's not a gas, but yeah. Now, so you can write methane, ammonia. Okay, over here it is mentioned. You have methane, you have ammonia, you have H2, also carbon dioxide, and also H2O, that is water vapor. It's not in the gaseous state, but still works. So instead of CO2, if you want to miss CO2, you can miss CO2. Rest of things you can write it over here. Hydrogen, ammonia, methane and H2O. Now, what conditions of primitive earth was recreated in the flask? High temperature and reducing environment. There was no oxygen. High temperature as in the flask, 800 degrees Celsius was being maintained. 800 degrees Celsius was being maintained. Now, write the conclusion drawn from this experiment. That basically life originated from inorganic things. These inorganic things came together to form organic compounds, and these organic compounds came together to form what the present day life forms that we see. Okay, next thing. List any 4 areas which provides evidences for evolution. First is paleontological evidences, my dear children, where there's fossils that gives us evidence. For example, I die. My bones will be still there. And on my bones there will be rock and things, there will be so many layers coming up, okay. And after so many years, probably when we humans are not there, if they dig and they come to this thing layer, they see, oh my god, what is this? This probably is not the thing that I am looking for. This is something like humans. There existed human beings, just like how we say there existed dinosaur, my dear children, just like that they will say there existed human beings. So this is paleontological evidence of evolution, that probably any organism that will be coming from me will be coming through me, like I am the source of evolution. I have evolved from the organism which were below me. You get it? So this is going to act like evidence. So fossils show gradual changes in organisms over time, linking extinct species to modern ones. Now, comparative anatomy. Okay, comparative anatomy, as in, for example, if you talk about cheetah, if you talk about me, cheetah and me, both of them have 4 limbs, okay, but they use their 4 limbs to run. I use my 4 limbs to do whatever I want, okay. So over here, if you see the 4 limbs, my dear children, it has humerus, radius, ulna, what do you call it? I forgot. This happens. Okay, I am tired now. So you have phalanges and all this thing. Okay, 4 phalanges, metacarpals and carpals. Yes. So the same thing is there in the cheetah also. So probably we guys might have evolved from what? The same ancestor. This is comparative anatomy. I am comparing my anatomy with their anatomy, and I am able to find that basically we both might have evolved from a common ancestor. Okay, so homologous and analogous organs suggest common ancestry or adaptive evolution. We are going to study about homologous and analogous later on. Okay, so comparative anatomy is the thing. Third thing, embryological evidence, my dear children. If you see our embryos, have you seen our embryos? How will you see our embryos? You might have heard that we had tail in the embryo. When we there in the womb of the mother, we have tail, we have gill slits and everything. Do we have tail now? Everyone check, please. Do we have tail now? We do not have tail now. Where does this tail go? Where basically when we get into adult stage, this tail is being lost. So who had this tail? Who had this tail? Amphibians had it, and all the other organisms have it, but not us. So probably we might have evolved from them. Who have the gill slits, my dear children? It's the fishes. It's the fishes that have it, but we also have it now. So what do you mean? Basically that shows that probably since we have the similar development patterns, probably we also might have evolved through all these organisms which are there. This was put down by Ernst Haeckel. Okay, embryological evidence was put down by Ernst Haeckel. I am explaining you one more question over here. Ernst Haeckel. But then there was another person who disapproved it. It was Ernst von Baer. It was Ernst von Baer who disapproved this theory by saying that it's just the fishes and amphibians that we show, not all the organisms which are there. And there's one last evidence, and that is molecular biology. That means similarities in our DNA, the proteins and the genes which are there, that is also going to act like one big evidence. Fine. So analyze the evolutionary relationship between forelimbs of whales and mammals. So I was talking about whales and mammals, my dear males and human beings. Sorry, mammals. Whales and humans. Listen, whales and human beings, both of us are mammals. Do you agree with that? Yes, you have to agree. Now, both whales and us have the same set of bones. Humerus, radius, ulna, carpals, metacarpals, phalanges. That shows that we have a common ancestry. But do we do the same thing? Whales basically they swim around with that. What do? We eat around, we dance, okay, with our hand. We do whatever we want. You get it? So that is the difference. Functionally we are different. Functionally we are different. But do we have same structures? Yes. We have same structural plan. Do we have common ancestry? Yes, we have common ancestry as well. So what do you call these type of structures as? You call this as homologous structures. And since two organisms are moving to different directions because they are staying in different habitat, because of that they have different functions, my dear children. This is known as divergent evolution. So they are diverging into the sea and we are diverging over here in the land, my dear children. So over here with my hand I just used to do this in this particular atmosphere. But whereas if we talk about water, in water if I go I have to use my hand for swimming. Just like that they are going to use their forelimbs for what? Swimming out there. Okay, so homologous organs, the forelimbs of whales and humans have same basic skeletal system but different function. Common ancestry, both evolved from a common tetrapod ancestor. Yes, tetrapod isn't the one does forelimbs. Okay, now showing divergent evolution. Different functions. Whales forelimbs modified into flippers for swimming, whereas human forelimbs is used for grasping and manipulation. Now same bone structure, both have humerus, radius, ulna, carpals, metacarpals and phalanges, but modified for different environments. That means we have different functions. This is written over here. So make sure you're also writing your answers properly. They're homologous structures. Why? Because they have common ancestry but they have different function and they have same bone function. So common ancestry, after that you can write same bone function, but then they have what? Different functions. So you same bone structure and then you write different bone functions. Different functions, that's all. Okay. Now my brain is going like this. Okay. Now state the evolutionary relationship giving reasons between sweet potato and potato. What is the evolutionary relationship between that? What do you call this type of organs as? So homologous organs, I hope it's clear. Homologous organs and divergent evolution, I remember like HD. So HD view of things. Okay. Now over here I'm going to talk about AC. So we are very rich. Okay, I'll tell you how. Sweet potato and potato, I'll say they are analogous organs. Why? Because suppose this is a tree share. This is a plant of potato. The potato stem basically grows up like this, enlarges to store things. Whereas when you talk about potato, okay, sorry, this is what? This is a potato. This is a potato where the stem is getting modified for storage of things. Now let's talk about the next thing, that is sweet potato. In sweet potato, the stem is there. If we talk about the roots are going to store a lot of things up there. The roots are the ones that are going to store a lot of things. So this is an example of sweet potato. Okay, where the roots are storing things. Now, both of their function, if I write functions, aren't they doing the same function of storing things? Yes. So function is same, but do they have same structure? Have they evolved from the same structure? No, it is from a stem, this is from a root. This is from a stem and this is from a root, my dear children. So it's not the same structure. Do they have common ancestry in that case? No. So these type of organs which have evolved together to have same functions because they are in the same environment, my dear children, you say that they are showing convergent evolution. They're converging. Now both of them have to store things since they are they are in soil. So that is why they have these things. So this is an example of convergent evolution. These organs are analogous organs. These organs are analogous organs. If you still didn't understand, let me give you another example. You have eye of octopus and the eye of human beings. Octopus is an invertebrate. They do not have a vertebral column. They're just animals, but they're totally different from us. But eye is there for watching, seeing things. Even here also in human beings, we watch things with our eyes, right? Or do we read things with our eyes? Do we do that? No, we read things with our mouth. We talk with our mouth, and we basically, even though we say philosophically that basically you are talking with your eyes and things like that, that is totally different. Yet all of us are basically seeing with our eyes, but structurally the eye of octopus and us is totally different. So we do not have any common ancestor. Again, example of what? Convergent evolution. Okay. So analogous evolutionary relationship between sweet potato and potato is that analogous structures. Sweet potato and potato have different origins but perform what? The same function of storage. Now different plant parts. Sweet potato is modified root for food storage, whereas potato is modified stem for food storage. Convergent evolution. They evolved independently to store food, adapting to similar environmental needs. Yes, they had similar environmental needs. That's why they adopted like that. No common ancestry. Despite similar function, they arise from different plant structures, showing adaptation rather than shared ancestry. Okay. Next thing. Mention the two key concepts of Darwinian theory of evolution. What are two concepts? One is natural selection. He said that organisms which has the favorable traits to survive, reproduce and pass on the traits, they will be the one that survive. The nature selects them. Okay. The less adapted one would not be selected by the nature. So the second thing, branching descent, that means descent with modification. New species evolve over generation due to gradual accumulation of variations from a common ancestor. Whatever new species that you see over here today, they have generated from the previous organisms which were there, and they have generated because of the gradual accumulation of what? Variations. Now this is not branching descent. New species are going to arise from generation to generation, my dear children, from the older ones. Okay, so all of them will be having a common ancestor. And natural selection. These are the two things, okay, the two key concepts of Darwinian theory of evolution. Now next thing, genetic drift is a chance while gene flow is constant. Substantiate. What do you mean by genetic drift and what do you mean by gene flow? We're going to talk about it when I talk about Hardy-Weinberg principle. Let's keep this question for the end. So question number 9 is for the end. Now, give any two examples of natural selection by anthropogenic action. What do you mean by natural selection? As in nature is selecting them. Okay, what do you mean by anthropogenic action? Human-made actions. Human-made disaster action, my dear son, that is anthropogenic action. So, what do you mean by natural selection and this no understanding, ma'am? So, basically, what did we do is that, suppose I had a fever. I started taking antibiotic for that. Okay. Now, slowly since I took so much antibiotics, my bacteria are all very resistant to these antibiotics that I am taking. So basically some of them, so there were initially Chutku, Butku and Kutku all over there with 4 different names. So I was taking the medicines usually. Slowly this guy realized, Chutku and Butku realized that we have to bring out some variation to be resistant to these chemicals. So they had this variation, they survived. Now every time I have this medicine, they will be surviving, other bacteria will be dying, but they will be surviving. So have I not made them resistant to certain drugs? Yes, I have, because I repeatedly took that, and now they have some changes in them. So this is anthropogenic action. I made it do. I made it happen, my dear children. Similarly, pesticides are also like that. I started putting pesticides. Now pests are resistant to this pesticide. They are like, I want to drink some pesticides. It's like that. Okay, even herbicides are also like that. So pesticides are for killing pests, herbicides are for killing, the organism that kills the herbs. Okay, so overuse of antibiotics, pesticides and herbicides has led to the evolution of resistant bacteria, insects and weeds, reducing the effectiveness of treatment and control measures. Apart from that, one famous example where human beings play a very important role is industrial melanism in moth. So you might have heard the story of moth. You have 2 moths, Biston metalliaria and Biston carbonaria, okay, the black moth and the white moth. So this is before industrialization. This is after industrialization. So before industrialization, the algae and the fungi used to form a combination, that is symbiotic association, that is known as lichen, my dear children, which were white in color. So they were spread all over the plants. Now this lichen is an indicator of pollution. That means whenever pollution is there, it won't be formed. Okay, so before industrialization, pollution was not there, so it was being formed. After industrialization, there was no lichen. There was no lichen which was being formed. Now you had white-winged moth. You had the white-winged moth, and you had the black one. Right? The white-winged one, when it came and sat over here, the prey which was there, the predator which was there, it was not able to spot it. Now if you think there is a black one sitting over here, you can easily spot the black one. You get it? So the white one increased in number, whereas the black one dropped in number. Here the nature selected the white one. Then because we did the pollution, we did everything, okay, we did industrialization, everything, pollution level raised up very much, okay. Now when the pollution level raised too much, my dear children, what happened is that the lichens were not able to form over here. The lichens were not able to form over here. The black ones, when they came and sat over here, no one was able to spot them. Okay, whereas when the white ones came and sat over here, all of the organisms could spot them and eat them. And this time the white ones level went down and this one level went up. So this is an example of natural selection with the help of anthropogenic action. So nature selected black ones. Why? Because of us, human beings. You get it? Because of our actions. Indirectly we are causing actions like that. So due to industrial pollution, darker moths had survival advantage over the lighter ones, leading to shift in what? Population, not pollution, population. Please remember, lichen is a natural indicator of pollution. Okay, very important question. Now, differentiate between homologous and analogous or structures. Give one plant and animal example for each. Homologous structures, structure with same origin but different function. I told you, structure with different origin but similar function is analogous. Divergent evolution, common ancestry is there, adapted for different use. Okay, convergent evolution, different ancestry there but adapted for similar functions. Now, thorns of bougainvillea and tendrils of cucurbita. You know, thorns which are there in bougainvillea and tendrils which is there in cucurbita. Okay, both of them, they are arising from the axillary bud. So they are arising from the same structure, right? So this is what common ancestry and common structure everything has, but they have different functions now. They have different function. Don't think the thorn looks different from tendril. Don't think like that. Basically, both of them came from the axillary bud. You have to think like that. So they have same structure and everything, common ancestry, but they do not have same function. So this is homologous structure. Sweet potato and potato, I told you, this is an example of analogous structure. Now in the case of animals, you have forelimbs of humans, whales and bats. Even bat is also mammal. Cheetah, all of them, my dear children, they have same structure but different function. Wings of butterfly and birds. You know, butterfly wings, you can put it like this. You should not be doing like that. Please don't do like that. I'm just giving an example, okay? So it's that brittle. But when I talk about birds, it is made up of feathers, bones and a lot of things up there. You get it? So they have different structure, but both of them are using it for what? Flying, my dear children. Same function of flying. So wings of butterflies and birds have different structure but same function of flying. So this is same function, that means convergent evolution, that means analogous structures. I hope you get it. Over here, vertebrate, heart and brain, everything an example of homologous structure. Vertebrate, heart and brain, even though they are not entirely similar, but they arise from the same structure and everything, since that is why it is being put over here. Please remember that, okay? Now, after industrialization in England, it was observed that white-winged moth did not survive. What is the cause? Because lichen was not there. After industrialization, soot from factories darkened the tree trunks, making white-winged moth more visible to predators, reducing their survival. And also, no lichen was there. No lichen was there, as they could not survive in high pollution thing. So what was the change and why it had happened? So dark moth increased because they blended better with the polluted tree. Right? This happened due to industrial melanism. This term is known as, this whole thing that is happening is known as industrial melanism, where dark colored individuals had a survival advantage, my dear children. Okay. Now, which organism is known as natural indicator of air pollution? It is the lichen. I told you, lichens are sensitive to pollution and disappear with areas with higher sulfur dioxide levels. Chalo, next. Australian marsupials and Australian placental mammals explain convergent evolution and adaptive gradation. Justify the statement. Okay, first things first. I have to talk about adaptive gradation. What do you mean by that? So, I am Darwin. I came into Galapagos Island for the first time. I saw finches, Darwin's finches, birds, okay. I was here, I was like, oh wow, so beautiful creatures. Then I moved to that side. When I went there, I found that same birds, but they have different beaks. They have different beaks and they were eating something else, insects and all. I went that side, I found another same birds, but again with more different beaks, and they were feeding on grass and everything. So I was like, oh my god, what happened? So probably what could have happened is, there was a common ancestor over here, birds. All of them, they went to different areas within the same geographical area, and they got better adapted there. This is known as adaptive radiation. So they are getting radiated first and getting adapted to that particular area. Now, when one such organism is moving and giving rise to different organisms, this is an example of divergent evolution, because common ancestor is there and they are going to different areas. Right, from a common ancestor you're getting different organisms which are having different functions, because they are living in different habitat. Here where there are insects are more, here where grasses are more, and things like that. So that is an example of divergent evolution. Same thing with Australian marsupials. So Australian marsupials were also there in Australian island. Okay, so these marsupials, they went to that side, this side, that side, this side, and they got to make so different type of organisms, such as kangaroos, bandicoots, sugar gliders, and everything. Again different areas, they were adapting better and they were forming different things. This is an example of, again, divergent evolution. Same thing with placental mammals. So we spoke about Darwin's finches, we spoke about Australian marsupials, and now we're talking about placental mammals. So placental mammals were also here, they were also radiating and making different things. All examples of divergent evolution. But if I'm telling you that in one location, that is in this particular studio, two different type of organisms are evolving. One from Australian marsupial, one from placental mammal, going to that side. Here also, they are moving to this side. Here also, they are moving to this side. These two guys which are going that side, they are not exactly same, but they will be having almost same function, because they are staying in the same habitat. Now these, do they have common ancestry? No, both of them have come from different organisms. This is an example of convergent evolution. So whenever two adaptive radiations happen in one particular place together, this is an example of convergent evolution. So you're asked about Australian marsupial and Australian placental mammals, that they show convergent evolution and adaptive radiation. What is this? You have to justify the statement. So adaptive radiation, because Australian marsupials evolved from a common ancestor into different species, adapting to various ecological niches. Okay, divergent evolution, such as kangaroo, bandicoots, and everyone. Convergent, why? Because, my dear children, placental mammals in other continents also evolved independently but developed similar adaptation as Australian marsupials. So example, marsupial anteater versus placental anteater. You know, both of them have come from different organisms, but they have similar feeding habitation. Okay. Thus, Australian marsupials show adaptive radiation, while the resemblance of two placental mammals from other regions is an example of convergent evolution. I can show you a picture. Where is that picture? I lost that picture. Just a second, okay? I think I lost a picture. I didn't put that over here. So sad. There was a picture, okay, which we had to follow. I don't know why. So you can also go check the adaptive radiation page, at the end, backside. For placental mammals, each placental mammal, you will be having one Australian marsupial as well. Okay. Now, State Hardy-Weinberg law lists 4 factors which disturb genetic equilibrium. Here I will be explaining you genetic drift and gene flow as well. The question which was there, question number 8 or something like that, right? So Hardy-Weinberg law says that the allele and genotypic frequencies in a population remain constant from generation to generation if no frequency forces act. So this is not there. If no evolutionary forces act, this is not there. Only this is there. That means there is no changes that is going to happen in the allelic and genotypic frequency in a population from generation to generation. For example, there is a generation of organisms where the amount of capital T allele is way too much than the small t allele. It is said that it will stay the same generation after generation. Do you think it can happen, my dear children? Do you think it can happen? It cannot happen. It cannot remain constant. But he called this as genetic equilibrium, and he put this in the form of a thing also, equation also, where he said p square plus 2pq plus q square is equals to 1. P represents the frequency of dominant allele. The frequency of dominant allele, for here I am taking capital T. Okay, q represents the frequency of recessive allele. The frequency of recessive allele, which is small t. Okay, now if I say that I want to find out the how many homozygous capital T organisms are there, what do I do? Whatever value of T is there, for example, if T is 0.2, then I will be doing 0.2 square, 2 times if I write TT. So that is P square. That is P square. P scores do not denote me that. Okay, fine, works. Now again I have to find what is the frequency of small t. Then suppose if this is 0.2, if p is 0.2, then what is q, my dear children? If p plus q is equals to 1, this is how we usually find out. Okay, so p plus q is equals to 1. If p is 0.2, then what will be q? What will be what will be q? Q is equals to 1.0.2, that is 0.8. Q is 0.8. So how do you calculate how do you calculate this? 0.8 square, that means q square. Now what is 2pq? For example, I want to find how many organisms are like this. So I am going to replace 2pq, that means p's value and q's value over here, then I will find out what is the frequency of this. This is how we need to find out, if at all numerical questions comes from this. Now my question is, do you think it will remain constant generation after generation? Suppose, okay, all of a sudden lots of short people decided to come to this particular place. This is La La Land. Okay, this is what? La La Land. To the La La Land, lots of short people decided to come. Okay, so there's too many small t alleles. What do you call this process as? You call this process as gene flow, my dear children. What do you call this? You call this process as gene flow. Gene flow is happening over here. Migration of individuals between population. Okay, this can naturally happen. Obviously, the genetic equilibrium can be changed. Now, suppose all the small people which are there, they decided to go off. Again, the gene flow will be happening, gene migration can happen. Next thing, suppose what happened? There was terrorist attack. There was some terrorist attack and they targeted all the tall people because they were not easy to hide. So, tall people's population went. This is a chance. Not always terrorist attack will happen. You get it? This is occurring by chance. What happened? All the tall people were being dead. Somehow, it's not like they were targeting tall people, but because they were seen, they were dead. Suppose if you don't understand that also, I can give you another example. There were some beetles, white beetles, and there were some green beetles. A big elephant came and stamped. Okay, it was walking like this. It stamped over here. All the white ones, accidentally they died. This is known as genetic drift. Random changes in the allelic frequency due to chance, my dear children, due to chance is known as genetic drift. Again, there is a population change, right? Now, next thing, suppose there was some mutation in this organism, there is some mutation, and over here, it is now giving rise to some red organisms. Again, the allelic frequency is going to change over here because of what? Mutations, sudden changes in DNA that creates what? Genetic variation. Next thing, natural selection. Because of something that is happening, the white ones could be spotted easily, they were killed. This is the nature's way of selecting them. Okay, favorable traits increase in frequency over the time. So, favorable traits for this one, they increase in frequency. The white ones, right? So, all these things are what? The factors that disturbs genetic equilibrium. There's one more since over here you are just asked to list any 4. It has not been mentioned. There's one more, and that is known as genetic recombination. That is known as genetic recombination. What do you mean by that? Basically, genetic recombination means in sexual interactions, suppose when chromosomes are coming together and things like that, okay, or basically when meiosis happens, there is change in the chromosome things, okay, they exchange certain things, certain parts of them. This leads to different things being formed over there, again leading to differences in the genetic equilibrium. So, these are the reasons that you have to remember. Please remember the equation also, p square plus 2pq is equals to q square is equals to 1. Sorry, plus q square is equals to 1. Now, this can be expanded form of p plus q whole square. You get it? Now, for finding out this thing, you write p plus q is equals to 1. This is the general thing that you do. Okay? Now, arrange the following. Okay, there's a question that I'll ask. If suppose they are given homozygous dominant organisms are like frequency of this is 0.49 is what? 0.49 homozygous dominant. That means you're talking that capital T is equals to 0.49. Now, if you want to find T, what do? Frequency of T. You do square root of 0.49, that is what? 0.7, my dear children. So, please understand you have to solve the questions like this if some numerical questions comes from this. Okay. Now the next question. Arrange the following ancestral forms of man in order to their evolution, So I'll tell you first. Okay, first you had Dryopithecus and Ramapithecus. 15 million years ago, Dryopithecus and Ramapithecus were there. Dryopithecus were more ape-like, Ramapithecus were more man-like. Okay, Rama, you can remember like that. Then from them, you have so many things happening, okay, in between. Then from them came Australopithecus. So first these two guys went off, okay. Then straight after 2 million years ago, we could find Australopithecus, which were more like, they had human-like features, they hunted with stones and all. They were tool makers, they hunted with stones and things like that. In the same era, from Australopithecus, we believe that Homo habilis developed. We think that in the same time period only, we see the fossils of some other organism which are known as Homo habilis. And we say Homo habilis were like the first ones to have hominid-like features, human-like features, totally human-like features. So Habibi, you can remember like that. Homo habilis as in the first Habibi that we had. And from Homo habilis, we got Homo erectus. So until Homo habilis, everyone were the ones who didn't eat vegetarians. They were like, no, I'm not going to eat non-veg food. I will only eat vegetarian food, only fruits and things like that. From Homo erectus, they started what? They started eating meat, my dear children. Okay, they hunted, they had very good tools and they started eating meat. After that, from them came Neanderthal man. Now, Neanderthal man, when they came, they were like, "You have not wore anything. I'm so shy." So they started protecting their body, and also they had some emotional connect. They started burying their dead as well. That is Neanderthal man. And parallelly, Homo sapiens, that is modern day Homo, us. Okay, they were also evolving. That is what is given in your NCERT textbook. I hope that is clear. I have an answer written over here. Okay, which we have to read, which we'll definitely read. Let's read it first. So, first in Dryopithecus and Ramapithecus, 15 million years ago, Dryopithecus were more ape-like, walked on all four limbs. Okay, Ramapithecus were more like man-like. They walked upright. At least they walked like this, not completely like this. They walked completely like this. Now, Australopithecus, 2 million years ago, lived in East African grasslands, walked upright, around 4 feet tall, hunted with stone weapons, but primarily ate fruits. What? They primarily ate fruits. Then came Homo habilis 2 million years ago. The first human-like hominid. Brain capacity was almost, this is very important, from here the brain capacity starts. The first human-like hominid, the brain capacity was 650 to 800 cc, my dear children. Used stone tools but did not eat meat. They also didn't eat meat. From here stone weapons started, but they primarily ate fruits. Here you add one more thing, that is brain capacity, that is 650 to 800 cc, cranial capacity, okay. Used stone tools but did not eat meat. Now, Homo erectus came almost around 1.5 million years ago. Fossils were discovered in Java also, okay, and the brain capacity was 900 cc. So, from 650 to 800, it became 900 cc. They started eating meat, as I told, and they started walking fully upright like this. Homo erectus, they were totally erect like this. Until now, some were like this, okay, but now it started being like this, erectus. Now, Neanderthal man, around 1 lakh to 40 years ago, okay, 40,000 years ago, lived in Near East and Central Asia. So, they were all, this was all happening in Africa and all. Some organisms, they went to Asia, Central Asia, and they're like, wow, so nice. Let me cover my body. So, it used hides for protection and buried their dead over here. Now, Homo sapiens, they evolved around this particular time, okay, between 75,000 to 10,000 years ago. They originated in Africa, spread worldwide, okay, developed into distinct races. Cave art and everything emerged around 18,000 years ago. That is not needed over here. So at least this thing, if you know, it's more than enough. Please remember, from this particular point, from Homo habilis, we start talking about the cranial capacity. Okay, before that we did not talk about the cranial capacity, and here 14,000 cc is very much important. Not 14,000, 1400 cc is very much important in Neanderthal man. In Homo sapiens also they have not mentioned, but in them they have mentioned. Now, a gene locus has 2 alleles, capital A, small a. If the frequency of dominant allele A is 0.4, I told you, right? Then what will be the frequency of homozygous dominant, heterozygous dominant and recessive individuals in the population? So, homozygous dominant, as in you're talking about, oops, AA is given, right? So, homozygous dominant, as in you're talking about capital A, capital A. Heterozygous dominant, as in talking about capital A, small a. And recessive individuals, as in talking about AA. What is given is that frequency of allele A is 0.4, and we know that this is dominant. So, P plus Q is equals to 1 in that. If we replace 0.4 plus Q is equals to 1, what is Q? 1 minus 0.4, then Q is 0.6. Q is 0.6. Okay, so where do we need 0.6? Over here. For finding AA, we calculate Q square. And what is Q? Q is 0.6. So Q square is 0.6 square, my dear children. What is 0.6 square? 0.36. Okay, that is homozygous recessive. What about homozygous dominant? Is there something that is known as homozygous recessive? Or every time if recessive comes, then it is homozygous only. In recessive condition only, the homozygous condition only, the recessive ones will be showing up. Now, homozygous dominant, here you want to find P square. P square, my dear children. How do you find that? 0.4 whole square is equals to P square. So what is it? 0.16. Now, heterozygous. That means AA. So here the children usually what do they do? Q and P, they just multiply. Please don't know that. You have 2 also. 2PQ. A plus B whole square is equals to A square plus 2AB plus P square. Right? So over here 2PQ. Please remember that. So 2 into 0.4 into 0.6, that is what? 0.48. So please write your answer like this. Will you? Yes, you should. And then the opposite is given, as I said, if suppose opposite is given, that means 0.36 is the frequency of homozygous recessive. What is the frequency of the allele? Small a. So what do you do? Square root of this, that is 0.6. Okay? Now, classify the following examples as homologous or analogous, and with this, we are going to complete all our questions. Are you excited? I hope you are. Chalo. So, eyes of octopus and mammals. Both of them are using it for viewing things, but do they have common ancestor? No. So, I will say this is convergent evolution, my dear children. Do I have? Oh no. I have to just say analogous or homologous, right? So, I will say this is analogous, not having same structure but having common function of seeing things. Wings of butterfly and birds. Again, analogous, they have the common function. Forelimbs of cheetah and bat. Cheetah and bat, cheetah use it for running, bat use it for flying. Is this common function? No. I will say this is homologous. Flippers of penguins and dolphins. Flippers of penguins and dolphins. This could confuse you. Penguin is a bird, but okay, please remember, penguin is a bird, dolphin is a mammal. So flippers, both of them have same function. Flippers of penguins and dolphins means we are talking about the same functions, right? So if same functions, then it is analogous. Thorns of bougainvillea and tendrils of cucurbita. Thorns of bougainvillea and tendrils of cucurbita. Please children, do not write, if flippers of penguins and dolphins, if flippers is the same for both, then it is analogous. Then forelimbs of cheetah and bird, then that is also analogous. Don't write like that. Okay, cheetah and bird, basically they are belonging to mammals. Have the basic understanding. Penguin is a bird, dolphin is not a bird. Okay, dolphin is a mammal. So obviously they are different ancestors are there, but they have common function, that is having flippers for swimming. Now thorns of bougainvillea and tendrils of cucurbita. Thorn, as I said, from axillary bud. Tendrils also from axillary bud. So same structure but different function. I'll call them homologous. So you should write your answer like this. Analogous, analogous, homologous, analogous. I hope this is correct. This is analogous, this is analogous, this is homologous, this is analogous, this is homologous. Yes. And let's see that question also about genetic drift. Don't tell that, ma'am, they'll tell the answer. Genetic drift, it is a random change in allele frequency due to chance events like natural disaster or isolation. More significant in small population and may lead to loss or fixation of the alleles. Yes, right? It is more significant in small population. You can clearly see the difference. Gene flow. Movement of alleles between population due to migration occurs continuously as individuals move between places and are interbred, increasing genetic diversity. Helps maintain genetic variation within the population. Genetic drift is unpredictable. Gene flow occurs constantly through migration. Works. I do not have a thank you slide, but I'll just say thanks to you, my dear children. So I hope this particular video, even though it's long, it's very, very important for you. Make sure you go through each and everything that I am telling you. Okay, don't miss anything. And I have given you a plan. Please follow that plan. In that way you can cover up all the videos, and by my videos itself I'm telling you, you'll get a lot of information which is there in the NCERT. But still recommending you to read the NCERT at least once. Use just 30 minutes of your time to read the NCERT. Okay, not beyond that, not going beyond that. Okay, if you can read it below that time, it's very good, but not beyond that. Okay, not have time to invest over that. Just understand the concept, do questions. It will be really helpful for you. If you found this video helpful, please let me know in the comment section, and lots of love to you. You are going to ace it. Okay, so bye bye, take care.